Close your eyes and breathe. Imagine the world above falling silent, the wind slowing, the stars dimming. Tonight, we aren't looking at the sky. We are descending, beneath your feet, into the quiet darkness of the soil, where something ancient is stirring. Something so small you could never see it with your naked eye, yet so vast it might stretch for miles beneath a single forest floor. This is the hidden kingdom, the silent architecture of life that connects every tree, every leaf, and every stone. Becomes even softer, more atmospheric. This is where we begin our journey tonight, into the hidden world of mycelium. 100 secrets of the earth, shared in the deep hours of the night. Let us start at the very beginning, with a single, microscopic spore. Beneath your feet, in the quiet darkness of the soil, something ancient is stirring. Something so small you could never see it with your naked eye, yet so vast it might stretch for miles beneath a single forest floor. This is where we begin our journey tonight, into the hidden world of mycelium. Let's start at the very beginning, with something almost impossibly small. Mycelium begins its life as microscopic spores. These spores are the seeds of the fungal world, though they're not seeds at all in the traditional sense. They drift through the air, settle on surfaces, wait in the soil, sometimes for years. And when conditions are just right, when moisture and temperature and nutrients align, these tiny capsules of potential awaken. They germinate. From each spore, a thread-like structure emerges, so delicate, so fine, that it exists at the very edge of visibility. This thread is called a hypha. And this is where the magic truly begins. Think about the patience required for this moment. A spore might land on a forest floor in autumn, lie dormant through the winter's freeze, remain still through the dry days of spring, and finally, when summer rains arrive and temperatures warm the soil, only then does it stir to life. This waiting is not passive. The spore is protected by incredibly resilient walls, designed to withstand extremes of temperature, desiccation, even digestion by animals. When germination finally occurs, it's like watching a time-lapse of a seed sprouting, except at a scale so small you'd need a microscope to witness it. The spore swells slightly, absorbs water, and then, a tiny protrusion appears. This is the germ tube, the very first hypha, reaching out tentatively into its new world. Imagine, if you will, a single hair-thin thread reaching out into the darkness. The hypha extends from what we call an apical tip, the growing point at its very end. This tip is alive with activity, constantly probing, sensing, reaching forward into new territory. At the cellular level, this tip is a marvel of biological engineering. Tiny vesicles, little bubbles of cellular material, stream forward from the body of the hypha toward this growing point, delivering the building blocks needed for expansion. The cell wall softens just enough at the very apex to allow the structure to push forward, then immediately strengthens behind the advancing tip to maintain the integrity of the tube. It's a continuous process of controlled growth, happening at a pace that might seem slow to us but is remarkably rapid at the cellular scale. And as it grows, something remarkable happens. The hypha doesn't simply grow longer in a straight line. No. It branches. It divides. It sends out new tips, new exploratory threads that themselves will branch again. What emerges is a pattern that nature loves, a fractal pattern, like the branches of a tree, or the tributaries of a river system, or the branching pathways of your own lungs. or the intricate tracery of lightning across a dark sky. Each new branch creates more branches, and those create more still, spreading outward in an intricate, tree-like colony that fills whatever space it inhabits. This branching is not random, it's purposeful, efficient, a living network designed to explore and exploit every available resource. When a hyphal tip encounters an obstacle, it doesn't stop. It grows around it, over it, sometimes even through it. When it senses nutrients in a particular direction, more branches form toward that source. When competition from other organisms is detected, the mycelium can redirect its growth, or even produce defensive compounds. This is intelligence of a sort, not consciousness as we understand it, but a sophisticated response system that has been refined over hundreds of millions of years of evolution. Now, let's pause for a moment and consider the scale of these structures. Each individual hypha, each of those thread-like filaments, is almost unimaginably thin. We're talking about tubular structures that measure between 1 and 30 micrometers in width. To put that in perspective, a human hair is roughly 70 micrometers across. So even the widest hypha is less than half the thickness of a single strand of your hair. Most are far, far thinner than that. At the smallest end of the scale, at just 1 micrometer, a hypha is so fine that it could slip between soil particles, through the tiniest cracks and crevices, penetrating spaces that nothing else can reach. Consider what this means for a moment. Soil is not solid, it's a matrix of particles with spaces between them, channels and pores created by the arrangement of mineral grains, organic matter, and the activities of countless organisms. Water flows through these spaces. Air fills them. And mycelium navigates them with extraordinary precision. A hypha 1 micrometer wide can thread through openings that would be impassable to bacteria clusters, plant roots, or larger soil organisms. This is part of mycelium's secret power, its ability to infiltrate, to move through the invisible gaps in the world, to access resources that other organisms cannot reach. The structure of a hypha is elegantly simple yet remarkably strong. It's essentially a tube, a cylinder with walls made primarily of chitin and other polysaccharides, filled with cytoplasm containing all the cellular machinery of life. Some hyphae are septate, meaning they have cross walls dividing the tube into compartments, each technically a separate cell. Others are cenocytic, forming long continuous tubes with multiple nuclei but no internal divisions. Both strategies have their advantages, and different fungal species have evolved different approaches to this fundamental architecture. And yet, despite their microscopic width, these hyphae can achieve extraordinary lengths. A single hypha can grow from just a few microns, thousandths of a millimeter, to meters long. Let that sink in for a moment. Meters. A thread thinner than anything you can see with your naked eye, stretching as long as you are tall, or longer, much longer. Some individual hyphae have been measured at several meters in length, and there's no theoretical limit to how long they might grow given adequate resources and space. Imagine that for a moment, really visualize it. Picture a filament so fine you'd need magnification to confirm it was there at all, yet extending across a room, or down through layers of soil, or through the length of a fallen tree trunk. It's almost paradoxical, this combination of delicacy and reach, of fragility and extent. And remember, we're not talking about just one thread. We're talking about thousands, millions, billions of these threads, all growing simultaneously, all branching and rebranching, creating a network of such density and complexity that it staggers the imagination. In optimal conditions, this network can grow several centimeters per day. That might not sound particularly impressive until you remember that this growth is happening simultaneously across millions of hyphal tips, each one extending, each one branching, each one adding to the total biomass and reach of the colony. The fastest growing mycelium can add several inches to its diameter in a single day, a silent explosion of growth happening in the darkness beneath our feet or within the wood of dead trees. 1. Where does all this growth happen? Mycelium spreads unseen beneath soil, wood, and stone. It lives in the spaces we don't think about, the hidden places. Under the forest floor, between particles of earth, within the decaying matter of a fallen log, through cracks in rock where moisture has collected and organic material has settled, in the gaps between tree roots, throughout compost heaps, inside the hollow spaces of rotting stems and branches. The mycelium moves through all of these spaces like a ghost, like water seeping through sand, like roots but far more delicate, invisible but omnipresent. Think about a forest floor for a moment. The surface layer you can see, the leaves, twigs, needles, and plant debris, that's just the beginning. Beneath that lies the organic layer where decomposition is actively occurring, and beneath that, the mineral soil. In each of these layers, mycelium is present, often in astounding quantities. Scientists have found that in healthy forest soils, the top few inches can contain several tons of mycelial biomass per acre. Tons of these microscopic threads, packed so densely and extending so extensively that their collective weight rivals or exceeds the weight of all the plant roots in the same volume of soil. You could walk through a forest where every single footstep lands on mycelium, tons of it, acres of it, spreading through the top layers of soil, and never see a trace of it. This is the secret kingdom beneath our feet, thriving, growing, connecting, all while remaining hidden from view. It's been called the Wood Wide Web by some scientists, and the nickname captures something essential about it, this vast, interconnected network that links the forest together in ways we're only beginning to understand. When we do catch glimpses of mycelium, perhaps when we peel back bark from a rotting log, or carefully sift through rich compost, or break open a mushroom to see the dense network at its base, what do we see? That filamentous microstructure most often appears white or cream in color. Sometimes it looks like fine cobwebs stretching across a surface, delicate and easily disturbed by the slightest breath of air. Other times it appears as dense mats, like felt or cotton wool spread across wood or soil. Sometimes it forms thick cords called rhizomorphs that look almost like roots, though they're still made of countless individual hyphae bundled together for strength and efficiency. This pale coloration isn't merely aesthetic, it's functional. These white to cream-colored threads are perfectly designed to penetrate substrates, to work their way into and through whatever material the fungus is colonizing. The color comes from the chitin in the cell walls, the same material that makes up the exoskeletons of insects and the shells of crustaceans, combined with other proteins and compounds that give the structure its strength and flexibility. Chitin is one of nature's most successful materials, second only to cellulose in terms of how much is produced by living organisms each year. This light appearance also reflects mycelium's nature as something that typically lives in darkness, without need for pigmentation to protect against sunlight. There's no chlorophyll here, No photosynthesis happening in these threads. Mycelium doesn't need to capture light, it captures something else entirely. It breaks down complex organic molecules and absorbs the simpler compounds that result. It's a decomposer, or recycler, returning nutrients to the soil and making them available for plants and other organisms. In darkness, beneath bark or soil or leaf litter, color serves no purpose. The pale filaments are optimized instead for penetration, for enzymatic secretion, for absorption of nutrients. When mycelium encounters wood, for instance, those cream-colored threads don't just sit on the surface. They produce enzymes, powerful biological catalysts, that break down cellulose and lignin, the structural compounds that make wood so strong and resistant to decay. The hyphae then absorb the sugars and other nutrients released by this breakdown. Slowly, steadily, the solid wood softens, becomes spongy, eventually crumbles. What was once a mighty tree returns to soil, molecule by molecule, through the patient work of mycelium. So here we have the beginning, the invisible beginning. Microscopic spores, smaller than grains of pollen, that could drift past your face on a breeze and you'd never know they were there. Spores that wait with the patience of stone for exactly the right moment to germinate. Hyphae that emerge from these spores and branch into fractals, creating patterns that echo throughout nature, in rivers, in trees, in the vessels of your own body. Threads so thin they slip through spaces we cannot see, navigating the microscopic architecture of soil and wood, yet so long they could stretch across rooms, through logs, beneath entire forests. A network spreading unseen through soil and wood and stone, appearing pale and delicate when we finally glimpse it, like frost patterns on glass, like spider silk and morning dew, yet powerful enough to break down the strongest materials on earth. Powerful enough to dissolve the lignin that makes wood hard, to decompose the cellulose that gives plants their structure, to recycle the very substance of the forest itself. This is mycelium in its earliest form, small, mysterious, and profoundly alive. Born from spores that drift and wait, growing through threads that branch and spread, building networks that connect and transform. It exists in the spaces between things, in the darkness beneath, in the hidden places where decay and renewal are one and the same. And we're only just beginning to understand how truly extraordinary it is. We're only just starting to glimpse the complexity of these networks, the sophistication of their growth patterns, the crucial role they play in nearly every terrestrial ecosystem on Earth. Let's continue our journey deeper into the hidden architecture of the fungal realm. Beneath the forest canopy, in the cool dimness of the soil, exists a structure so elegantly simple yet so profoundly complex that it challenges our very understanding of what it means to be an organism. We've spoken of mycelium's beginnings, its microscopic origins and spreading growth. Now, let us look more closely at what mycelium actually is. What are these pale threads made of? What gives them their strength, their flexibility, their remarkable ability to persist and thrive in environments that would challenge almost any other form of life? The answer begins with a simple truth. Mycelium consists entirely of interwoven hyphae. That's it. That's the fundamental architecture. There are no specialized organs, no distinct tissues in the way we think of them in plants or animals. Just hyphae. Threads upon threads, weaving together, overlapping, branching, rebranching, creating through their collective arrangement all the structure and function the organism needs. It's a bit like understanding that a rope, no matter how thick or strong, is ultimately just fibers twisted together. But unlike a rope, mycelium is alive, every thread, every filament, actively growing, responding, changing. This simplicity is deceptive. Within that apparently straightforward structure lies extraordinary complexity. The way these hyphae interweave isn't random, it's organized, purposeful. In some areas, they form loose networks with space between threads, allowing for maximum exploration and resource capture. In other areas, they pack tightly together, creating dense structures that can push through resistant materials or form the protective outer layers of fruiting bodies, the mushrooms we see above ground. This same basic building block, the hypha, arranged in different densities and orientations, creates all the functional diversity the organism requires. Let's zoom in closer now, to the level of a single thread. Each hypha forms a single elongated cell or chain. Now, this requires a moment of explanation, because fungal cells don't always behave the way we expect cells to behave based on what we learned about plants and animals. In some fungi, each hypha is divided by cross walls called septa, creating a chain of individual cells lined up end to end, like beads on a string or railroad cars in a train. These septa aren't completely closed. They have pores that allow cytoplasm, organelles, and even nuclei to flow between compartments. So while technically divided into cells, the hypha still functions as a connected unit, sharing resources and information along its length. In other fungi, the hyphae are cenocytic, meaning they lack these cross walls entirely. Imagine a single cell stretched out like taffy, extending for centimeters or even meters, with no internal divisions. These cenocytic hyphae are essentially long tubes of cytoplasm containing multiple nuclei, all functioning within one continuous cellular space. Both strategies work. Both have evolved and persisted. The septate hyphae can compartmentalize damage. If one section is injured, the septa can seal off and protect the rest of the filament. The cenocytic hyphae can move resources more freely along their length without the slight resistance that septa create. Nature, as always, finds multiple solutions to the same problems. What gives these delicate threads their structure, their ability to maintain form and push through resistant substrates? The answer lies in their construction. Hyphal walls are rigid and chitin-based. Chitin, that remarkable polysaccharide, that nitrogen-containing compound that nature has used over and over again in different contexts. It's what makes the shells of beetles hard, what gives structure to the exoskeletons of spiders and lobsters, what stiffens the wings of butterflies. And here, in the fungal kingdom, it forms the primary structural component of hyphal walls. Chitin molecules are long chains of N-acetylglucosamine, linked together in a configuration that creates crystalline microfibrils. These microfibrils are incredibly strong. Pound for pound, chitin rivals steel in tensile strength. When woven together into the matrix of a hyphal wall, embedded in a cement of proteins and other polysaccharides, they create a structure that is simultaneously rigid enough to maintain the tube shape of the hypha and flexible enough to bend without breaking. This is crucial for an organism that must navigate through soil particles, around roots, through the grain of wood, constantly encountering resistance, constantly adapting its path while maintaining structural integrity. The rigidity of these chitin-based walls allows hyphae to generate turgor pressure, the internal hydraulic force that drives their growth. Water flows into the hypha, increasing pressure against the walls, and this pressure pushes the growing tip forward into new territory. Without rigid walls to contain this pressure, such growth would be impossible. The hypha would simply balloon outward rather than extending forward. It's engineering at the cellular level, a perfect marriage of material science and biology that has been refined over hundreds of millions of years. Inside these chitinous tubes, life continues in all its microscopic complexity. Hyphae contain cytoplasm with multiple nuclei. This is another unusual feature of fungal biology. While most plant and animal cells contain a single nucleus, the control center housing the organism's DNA, fungal hyphae often contain many nuclei, sometimes dozens in a single hyphal compartment. These nuclei may even be genetically different from each other, a condition called heterokaryosis. Imagine a single cell functioning as a genetic democracy, with multiple slightly different sets of instructions all being expressed simultaneously. This can provide tremendous adaptive advantages, allowing the fungus to respond to environmental challenges with a diversity of genetic responses without waiting for mutations and selection to occur over generations. These multiple nuclei drift through the cytoplasm, moving along with the flow of cellular contents toward growing tips where new cell wall material is needed, or redistributing themselves after cell division. They're not fixed in place like the nucleus in a human cell, anchored by cytoskeletal elements. They're mobile, dynamic, responding to the needs and conditions within the hypha. It's a fluidity that reflects the overall character of the mycelial network, always adapting, always redistributing resources to where they're most needed. But nuclei are just the beginning of what these hyphae contain. The cytoplasm is crowded with the machinery of life. Ribosomes, mitochondria, vacuoles, and globules power the cell. Each of these organelles has its essential role to play. The ribosomes, tiny molecular machines made of RNA and protein, are where the genetic information carried by the nuclei is translated into actual proteins, enzymes, structural components, regulatory molecules. Proteins are the workhorses of cellular function, and ribosomes are their factories, churning out these essential molecules in response to the cell's needs. The mitochondria are the powerhouses, the energy transformers of the cell. These remarkable organelles, with their own DNA and their own evolutionary history, take the sugars and other nutrients that the mycelium has absorbed and convert them through cellular respiration into ATP, adenosine triphosphate, the universal energy currency of life. Every function the hypha performs, from synthesizing new cell wall material to actively transporting nutrients to generating the enzymes that break down substrate, requires energy. The mitochondria provide it, burning fuel and controlled reactions that capture energy in usable form. The vacuoles deserve special attention because of their prominence in fungal cells. These membrane-bound compartments serve multiple functions: storage, waste disposal, maintaining turgor pressure, breaking down old cellular components for recycling. In plant cells, vacuoles can be large, but in fungal hyphae, they can be truly dominant. Vacuoles can occupy up to 90% of hyphal volume. Think about that proportion for a moment. If a hypha is like a tube, imagine that tube almost entirely filled with these fluid-filled sacs, leaving just a thin layer of active cytoplasm pressed against the cell wall. It seems counterintuitive. Why would a cell dedicate so much space to what are essentially storage compartments? The answer lies in efficiency and function. Those vacuoles are filled with water, and that water generates the turgor pressure that drives growth and maintains structure. They're also storage facilities for nutrients, amino acids, ions, supplies that can be drawn upon when needed. They contain degradative enzymes that break down materials the cell needs to recycle. And by occupying so much volume, they actually allow the hypha to be extremely economical with its metabolically active cytoplasm. A thin layer of living material surrounding a pressurized, fluid-filled core. It's an elegant design that minimizes the energetic cost of maintaining cellular functions while maximizing volume and therefore pressure and storage capacity. The globules mentioned are lipid droplets, stores of fats and oils that represent concentrated energy reserves. When conditions are favorable and nutrients abundant, the mycelium stores excess in these lipid globules. When times are harder, when growth is moved into nutrient-poor substrate, these reserves can be mobilized, broken down to provide energy and building blocks for continued expansion. Now let's return to the physical dimensions of these structures. We mentioned earlier that hyphae can range from 1 to 30 micrometers in width, but that's a general range. When we look more specifically at the actual diameter of the cytoplasm-filled interior of hyphae, we find that hyphal diameters vary from 0.2 to 3 micrometers in many species. The smallest of these, at 0.2 micrometers or 200 nanometers, are almost incomprehensibly fine. For reference, that's roughly the wavelength of violet light. These ultra-thin hyphae can penetrate the tiniest spaces, slip between soil particles that are only slightly larger than the hyphae themselves, explore microcracks and substrates that other organisms cannot access. At the larger end, 3 micrometer hyphae are still microscopic but substantially more robust. They can transport more nutrients, contain more nuclei and organelles, generate more enzymatic output. Different species have evolved different optimal diameters based on their ecological strategies. Some fungi specialize in fine, penetrating hyphae that maximize surface area relative to volume, important for absorbing nutrients efficiently. Others develop thicker hyphae that can transport resources over longer distances more effectively, connecting different areas of the mycelial network like highways connecting cities. The structure of the hyphal wall itself varies considerably. Wall layers range from 2 to multi-layered. At its simplest, a hyphal wall might consist of just 2 layers, an inner layer rich in chitin microfibrils providing tensile strength, and an outer layer containing proteins and other polysaccharides that mediate interactions with the environment. But many fungi elaborate on this basic plan. Some have 3, 4, or even more distinct layers visible under electron microscopy, each with its own composition and function. These multiple layers can serve various purposes. Outer layers might be specialized for secreting enzymes, for protecting against desiccation, for interacting with plant roots in symbiotic relationships, or for resisting attack by bacteria or other fungi. Middle layers might be particularly rich in chitin for structural support. Inner layers might be modified to regulate what passes between the cytoplasm and the wall, controlling water movement and the loss of cellular contents. The complexity of these wall structures reflects the complexity of the environments fungi inhabit and the challenges they must overcome. In these walls, these boundaries between the living cytoplasm and the external world, are not uniform in their appearance. Hyphae may be smooth, encrusted, pigmented, or colorless. Smooth hyphae have walls that are essentially featureless at the microscopic level, clean tubes of chitin and protein extending through substrate. These are perhaps the most common form, optimized for growth and penetration without unnecessary elaboration. Encrusted hyphae, however, have walls decorated with crystals, granules, or other materials precipitated onto their surfaces. Sometimes these are calcium oxalate crystals, byproducts of metabolism that the fungus deposits on its own cell walls. Sometimes they're mineral particles from the environment that become embedded in the wall material. These incrustations can serve protective functions or may simply be the result of the chemical environment in which the hyphae are growing. Pigmented hyphae contain melanins or other colored compounds in their walls. While we mentioned earlier that much mycelium appears white or cream because it lives in darkness where pigmentation isn't necessary, many fungi do produce pigmented hyphae. Browns, blacks, grays, and even yellows or greens can appear in hyphal walls. Melanin in particular is important. It provides protection against ultraviolet radiation, but also against enzymatic attack, against extreme temperatures, against desiccation. Dark, melanized hyphae are often found in fungi that grow in exposed conditions or that need to persist through environmental stresses. Colorless hyphae, of course, lack these pigments, remaining translucent or appearing white simply because of the way light scatters off the chitin structure. These are often the fastest growing, most metabolically active hyphae, investing their resources in expansion rather than in the production of protective compounds. They thrive in stable, protected environments where the benefits of pigmentation don't outweigh the costs of producing pigment molecules. So here we have the building blocks, the fundamental architecture of mycelial life. Interwoven hyphae, each one a cell or chain of cells, each protected by rigid chitin-based walls that can be simple or complex, smooth or textured, pigmented or clear. Inside these walls, cytoplasm crowded with nuclei, sometimes many, each contributing its genetic instructions to the collective function. Ribosomes building proteins, mitochondria generating energy, vast vacuoles maintaining pressure and storing resources, lipid globules holding reserves against uncertain times. Diameters ranging from impossibly fine to merely microscopic, walls layered in two or more distinct sheets, surfaces that can be smooth as glass or rough as sandpaper, clear as water or dark as earth. All of this variation, all of this complexity, arising from a single basic structure, the hypha. Tubes of life, extending, branching, weaving together into networks that can span forests, networks that connect trees, that break down the dead and return nutrients to the living, networks that have been fundamental to terrestrial life for hundreds of millions of years. This is the living architecture of mycelium, simple in concept but infinite in variation. This is what lies beneath every footstep in the forest, what fills the rotting log, what threads through the compost in the soil. These are the building blocks of a living network that connects and sustains the world above. In the stillness of the soil, something remarkable is happening. Without muscles, without nerves, without any of the machinery we typically associate with movement, mycelium grows. It extends, it reaches, it explores new territory with a purposefulness that seems almost deliberate. How does something so simple, so apparently passive, achieve such dynamic growth? How do these microscopic threads push through resistant substrates, navigate complex environments, and expand their networks with such efficiency? The answer lies in understanding how growth actually happens, in the precise choreography of molecular events occurring at the very tips of these fungal filaments. Here's something that might surprise you. Despite the extensive networks that mycelium forms, despite the millions of threads that make up a mature colony, growth occurs only at hyphal tips. Not along the length of the mature hyphae. Not from random points throughout the network. Only at the tips. Each growing tip is a site of intense cellular activity, a frontier where the organism literally constructs itself forward into new space. Behind the tip, the hyphal wall has matured, rigidified, become fixed in its final form. But at the tip, everything is fluid, dynamic, under construction. This concentration of growth at the tips is actually a brilliant strategy. It means the fungus can focus its resources, its energy, its cellular machinery on a relatively small number of actively growing points while maintaining a vast network of mature hyphae that serve primarily as transport highways and structural support. Imagine if you had to maintain every part of your body at the same high metabolic rate required for growth. It would be impossibly expensive in terms of energy. The mycelium avoids this by growing only at specific locations while the rest of the network operates in a more economical maintenance mode. But what exactly is happening at those tips? What makes them special? Deep within each growing hyphal tip, there exists a structure so tiny, so specialized, that it wasn't even clearly observed until electron microscopy revealed its secrets. This is the Spitzenkörper, a German word meaning apex body. The Spitzenkörper vesicle clusters control tip growth. Think of the Spitzenkörper as a command center, though of course it has no consciousness, no intentionality. It's a collection of tiny membrane-bound vesicles and associated organelles, clustered together just behind the very apex of the growing tip. Under the electron microscope, it appears as a dark, dense region amid the cytoplasm, a congregation of activity, a hub of construction. These vesicles are absolutely essential. They're like cargo containers, each one carrying the raw materials needed to build new hyphal wall at the advancing tip. The vesicles deliver chitin synthase to the growing wall. Chitin synthase is an enzyme, a molecular machine that takes simple sugar molecules and links them together into the long chains of chitin that form the structural framework of the hyphal wall. The enzyme itself is too large and too hydrophobic to simply float freely through the watery cytoplasm to where it's needed. Instead, it's packaged into vesicles, tiny bubbles of membrane that can be transported, directed, and then fused with the cell membrane at exactly the right location. Picture this process for a moment. In the cellular interior, perhaps several micrometers back from the tip, endoplasmic reticulum and Golgi bodies are busy synthesizing chitin synthase enzymes and other wall components, packaging them into vesicles. These vesicles bud off and begin their journey toward the tip, guided by the cellular equivalent of railroad tracks. They stream forward, joining the collection of vesicles in the Spitzenkörper. And then, in a carefully orchestrated dance, these vesicles move from the Spitzenkörper to the cell membrane at the very apex of the tip, fuse with that membrane, and release their contents. The chitin synthase enzymes embed themselves in the membrane and immediately begin their work, spinning out chitin chains that become part of the expanding wall. This is growth. Not the slow accumulation of material that we see in a crystal forming, but active construction, enzyme-driven synthesis occurring in real time at a moving front. The tip advances because new wall material is continuously being added to the apex, while turgor pressure, that internal hydraulic force, pushes the tip forward into the softened, newly synthesized wall. It's construction and expansion happening simultaneously, a moving factory that builds the road as it travels. The growing tip has a characteristic shape. A 1 to 2 micrometer apical dome forms at the tip. This dome is the region where the wall is newest, most recently synthesized, and therefore most flexible. Behind this dome, the wall gradually matures, additional layers are added, cross-linking between molecules increases, and rigidity develops. But the dome itself remains soft enough to yield to the internal pressure, to expand as growth occurs. The shape of this dome, its exact curvature, its dimensions, is actually determined by the pattern in which vesicles fuse with the cell membrane and deliver their contents. If vesicles fuse uniformly across the dome, the hypha grows straight. If vesicle fusion occurs preferentially on one side, the tip curves in that direction. This is how hyphae navigate, how they respond to environmental cues. Chemical signals from the environment, physical obstacles, even subtle variations in substrate density can influence where vesicles fuse, and therefore which direction the tip grows. It's a steering mechanism operating at the molecular level, allowing the mycelium to explore its environment with what almost seems like purposeful exploration. But how do those vesicles know where to go? How does the cellular machinery ensure that they move from their point of origin to the Spitzenkorper, and from there to the exact right location on the expanding tip? The answer involves another level of cellular architecture. Microtubules guide vesicle transport. Microtubules are hollow tubes made of protein, part of the cellular cytoskeleton, the internal scaffolding that gives cells their shape and organization. These microtubules extend through the cytoplasm of the hypha like tracks through a subway system, providing pathways along which vesicles can move. Motor proteins, molecular machines powered by ATP, attach to the vesicles and literally walk along the microtubules, carrying their cargo forward. It's active transport, energy-driven movement that's directional and purposeful. The microtubules radiate from organizing centers back in the hypha toward the Spitzenkorper at the tip, creating highways that naturally direct traffic in the right direction. Without this organized transport system, vesicles would simply drift randomly through the cytoplasm, and coordinated growth would be impossible. With it, the cell can deliver materials precisely where they're needed, when they're needed, in the quantities needed. This is all happening at a scale and speed that's difficult to fully appreciate. We're talking about vesicles a few hundred nanometers in diameter, being moved along tracks only 25 nanometers wide, traveling distances of several micrometers, which at their scale is like crossing several city blocks, all within seconds. And this is happening simultaneously for hundreds or thousands of vesicles, all moving forward, all contributing to the continuous growth of the tip. But vesicle transport isn't the only flow happening within hyphae. The entire interior of a hypha is in motion. Cytoplasm streams bidirectionally up to 1 centimeter per minute. 1 centimeter per minute. In a structure that's only a few micrometers wide, that's blazingly fast. Imagine water rushing through a pipe, or wind rushing through a narrow canyon. That's the equivalent speed, relatively speaking, of cytoplasmic streaming in hyphae. And this streaming isn't unidirectional. It's bidirectional, meaning flow can occur simultaneously in both directions. In the center of the hypha, cytoplasm might be flowing toward the tip, carrying nutrients, organelles, and vesicles to the growing frontier. Near the walls, return flow carries waste products, excess water, and recycled cellular components back toward older parts of the mycelium. This creates a circulation system, a way for the organism to move materials around without any pumping heart, without any muscle contractions, purely through the physics of fluid dynamics and the molecular motors that generate flow. Why is this streaming important? Because mycelium can extend for meters, and nutrients absorbed at one location might be needed somewhere far away. A hyphal tip pushing into fresh substrate might need amino acids that were absorbed 10 centimeters back where the mycelium broke down a rich deposit of organic matter. The streaming cytoplasm is the transportation system that makes such nutrient sharing possible, that allows the mycelial network to function as an integrated whole rather than as millions of independent threads. What drives this internal flow? Pressure gradients drive internal flow. Just as water flows downhill, from high pressure to low pressure, cytoplasm flows through hyphae following pressure differences. These pressure gradients can be generated by several mechanisms. Turgor pressure might be higher in regions where water is being actively absorbed, creating a pressure head that drives flow toward regions of lower pressure. Active transport processes might generate osmotic differences that pull water and dissolved materials in particular directions. The contraction of actin filaments, another component of the cytoskeleton, can generate local pressures that push cytoplasm along. The result is a self-organized system where flow naturally moves materials from regions of abundance to regions of need. It's the same principle that drives sap flow in plants, blood flow in animals, water movement through soil, pressure seeking equilibrium, with the movement itself serving the organism's needs. And because pressure can be modulated by cellular activity, the mycelium has a degree of control over where materials flow, allowing it to prioritize rapidly growing tips or to reallocate resources from slower growing regions to areas encountering richer substrates. This brings us to one of the most fascinating aspects of hyphal growth, its responsiveness. Branching responds directly to nutrient availability. When a hypha is growing through nutrient-poor substrate, it tends to grow fairly straight, minimizing branching, conserving resources, essentially exploring as rapidly as possible to find better conditions. But when a hyphal tip encounters a region rich in nutrients, perhaps a deposit of decomposing organic matter, perhaps the surface of a nutrient-rich material, something changes. The tip slows slightly, and behind it, new branches begin to form. These branches aren't random. They emerge near the site of nutrient abundance, and they proliferate in number. The result is what mycologists call foraging behavior. The mycelium responds to finding food by intensifying its exploration and exploitation of that resource-rich area. It's as if the fungus knows it's found something valuable and deploys more exploratory threads to make the most of the discovery. Of course, the fungus knows nothing in a conscious sense. But evolution has shaped its growth responses so that it automatically, mechanically, increases branching density where nutrients are abundant. The mechanism behind this involves chemical signaling within the mycelium. When nutrients are absorbed, They trigger cascades of molecular signals that affect gene expression, protein synthesis, and ultimately the organization of the Spitzenkorper and the pattern of vesicle fusion. More branch initiation points form. The cytoskeleton reorganizes to support multiple growing tips instead of just one. The single expanding front becomes many fronts, each exploring, each absorbing, each contributing to the overall network's growth and nutrient capture. This responsiveness extends to other environmental factors as well. Hyphae can grow toward moisture gradients, toward optimal temperatures, away from toxic substances, toward symbiotic partners. All of this happens through variations in growth rate, branching frequency, and growth direction, which ultimately all come down to the precise control of vesicle fusion at hyphal tips, the flow of cytoplasm through the network, and the pressure differentials that drive that flow. Think about what this means for the mycelial network as a whole. You have millions of tips, each one a semi-autonomous growing point, each one capable of sensing its local environment and responding appropriately. No central brain coordinates this. No master controller decides which tips should grow faster or branch more. Instead, each tip responds to its own immediate conditions, and from these millions of local decisions, coherent network-level behavior emerges. The mycelium as a whole appears to intelligently forage, to efficiently explore, to optimize its network architecture, not through top-down planning, but through bottom-up responsiveness. This is growth without muscles, navigation without nerves, behavior without brains. It's the choreography of molecules, vesicles streaming forward along microtubule tracks, enzymes embedding in membranes and spinning out structural polymers, cytoplasm flowing along pressure gradients, turgor pressure pushing tips into new territory. At the apex of each growing tip, in that 1 to 2 micrometer dome of soft, expanding wall, the future of the mycelium is being written, molecule by molecule, as chitin synthase builds the scaffold and the Spitzenkorper orchestrates the delivery of materials to the construction site. Behind each advancing tip, mature hyphae serve as supply lines, with cytoplasm streaming through them at remarkable speeds, redistributing resources, connecting the dispersed parts of the network into a functional whole. And everywhere, constantly, the mycelium responds to its world, branching where nutrients are found, growing straight where they're absent, turning toward favorable conditions, avoiding harmful ones, all through the exquisite control of growth at those critical points, the hyphal tips. This is how forests are woven together beneath the soil. This is how fallen trees are slowly, steadily returned to earth. This is how nutrients cycle, how carbon flows, how the biological and geological worlds interpenetrate. All of it resting on these fundamental processes: vesicle transport, enzyme activity, cytoplasmic streaming, pressure-driven flow, environmentally responsive branching. Simple mechanisms, repeated millions of times across millions of tips, creating networks of stunning complexity and effectiveness. This is growth at the frontier of life, construction happening in real time, the continuous, unceasing expansion of the mycelial network. Movement without muscles. Purpose without consciousness. The silent, persistent growth of the fungal realm. In the hidden world beneath our feet, mycelium faces constant challenges. A burrowing beetle larva tears through hyphal strands. A probing root tip ruptures delicate filaments. Frost crystals form and expand, shearing cellular structures. water drains away suddenly, leaving hyphae vulnerable to collapse. The underground environment is not gentle, not stable, not forgiving. And yet mycelium persists, thrives, endures across seasons and years and decades. How? What allows these fragile threads, these microscopic tubes that we've described, to withstand the mechanical chaos of the soil environment? The answer lies in internal architecture and damage control systems so elegant they seem almost engineered. Let's begin with a closer look at the internal structure of individual hyphae. We've mentioned this before, but now we need to understand it more deeply. Septa partially divide hyphae. These septa are crosswalls, thin barriers that span the width of the hyphal tube, creating compartments along its length. But here's what makes them remarkable: they're partial divisions. They don't completely seal off one section from another. Instead, they maintain the hypha as a functionally continuous structure while providing a kind of internal scaffolding, a series of support walls that strengthen the overall architecture. Think of septa like the bulkheads in a ship's hull. They divide the interior space, but they don't eliminate it. They provide structural reinforcement so that the long tube doesn't simply collapse under pressure or buckle when it encounters resistance. A hyphal filament might extend for centimeters or more, and without some internal support, such a thin-walled tube would be mechanically unstable. The septa provide that support, spaced along the hypha's length like the ribs along a submarine's pressure hull, maintaining the circular cross-section and preventing collapse. But unlike solid walls, septa in fungi have a special feature. Septal pores allow cytoplasmic streaming. At the center of each septum, or sometimes at multiple points across its surface, there are openings, pores that permit the passage of materials from one compartment to the next. These pores vary in size depending on the fungal species, but they're typically large enough to allow not just small molecules in cytoplasm, but even larger organelles to pass through. Mitochondria can squeeze through septal pores. Nuclei, in some species, can migrate from compartment to compartment. Vesicles, ribosomes, all the cellular machinery we've discussed, all of it can flow through the network of connected compartments. This creates a beautiful compromise. The hyphae gain structural integrity from the septa, which divide the long tube into shorter, more mechanically stable segments. But they retain functional continuity because cytoplasm can stream freely through the pores, maintaining the hypha as a unified living system rather than a chain of isolated cells. Resources absorbed at the growing tip can flow backward through septal pores to older parts of the mycelium. Nutrients stored in vacuoles in one compartment can be mobilized and transported through pores to where they're needed. The mycelium functions as an integrated network despite being physically divided. The size and regulation of these pores is actually quite sophisticated. In some fungi, proteins form complex structures around the septal pores, creating what's called a pore cap or septal pore apparatus. These structures can regulate flow through the pore, potentially controlling what passes and what doesn't, acting like molecular gatekeepers. The pore might be open during periods of active growth and nutrient transport, allowing free flow. But under certain conditions, stress, damage, or developmental changes, these regulating structures can restrict or even close the pores. Which brings us to one of the most remarkable aspects of septal biology. Imagine a hypha is damaged. Perhaps an insect mandible has severed it, or a sharp soil particle has punctured the wall. Without septa, the entire hyphal length would be compromised. Cytoplasm would leak out through the breach, pressure would drop, and the entire filament might die. But with septa and their associated damage control mechanisms, the story is very different. Wernin bodies rapidly seal septa after injury. Wernin bodies are specialized organelles unique to certain groups of fungi. They're roughly spherical structures, about 1 to 3 micrometers in diameter, composed of crystalline protein. Under normal conditions, they float in the cytoplasm near septal pores, tethered loosely by protein filaments, essentially just waiting. They're an emergency response system on standby. When damage occurs and a breach in the hyphal wall develops, the sudden change in pressure and the flux of cytoplasm toward the breach triggers the Wernin bodies into action. Within seconds, sometimes within less than a second, Wernin bodies nearest to the damaged compartment move to the septal pores and physically plug them. They're pulled by the cytoplasmic current toward the breach, and when they encounter a septal pore, their size and shape cause them to lodge in the opening, creating an instant seal. This prevents cytoplasm from the undamaged compartments from flowing into the breached compartment and being lost. Its damage control at the cellular level, automatic and remarkably fast. The breached compartment may be lost, its contents spilling out, its turgor pressure collapsing, its cellular machinery ceasing to function. But the rest of the hypha survives, sealed off by the Wernin body plugs, maintaining pressure, maintaining function. The mycelium can survive the loss of individual compartments, can endure localized damage, because this sealing mechanism prevents cascade failures. Its resilience through compartmentalization, survival through sacrifice of the damaged parts to preserve the whole. Not all fungi have Wernin bodies. They're found primarily in the Ascomycota, but those that lack them often have alternative mechanisms for sealing septa. Some produce rapid deposits of callose, a polysaccharide that can quickly gel and block pores. Others have contractile proteins that can constrict septal pores shut. The specific mechanism varies, but the principle is universal. Damage must be contained, compartmentalized, prevented from spreading through the mycelial network. Now, let's zoom out from individual hyphae and consider the network as a whole. We've been talking about mycelium as a network, but what exactly makes it a network rather than just a collection of branching threads? The answer is anastomosis. Anastomosis fuses hyphae into networks. Anastomosis is the process by which two separate hyphae, either from the same mycelial colony or, in some cases, from genetically compatible but physically separate colonies, recognize each other, grow toward each other, and fuse. Picture this happening in the soil. A hypha is growing, branching, exploring. One of its branches encounters another branch from the same mycelium, or from a nearby colony. Through chemical signaling, through detection of self or compatible non-self markers, the two hyphae recognize each other as potential fusion partners. Their tips orient toward each other. They grow together until they touch. Then, in a carefully controlled process, the cell walls at the contact point dissolve. The membranes fuse. The two hyphae become one continuous structure, with cytoplasm now able to flow freely between what were previously separate branches. This process transforms the mycelial architecture from a tree-like branching structure into a true network with interconnections, loops, alternative pathways. In a purely branching structure, there's only one path between any 2 points. You have to trace back through the branches to a common node, then forward again. But in a networked structure created by anastomosis, multiple paths exist between most points. This has profound implications for robustness and efficiency. If a single hypha in a tree-like structure is severed, Everything beyond that point is cut off from the rest of the mycelium. But in an anastomosed network, alternative pathways exist. Nutrients can be rerouted around damage. Information, in the form of chemical signals, electrical impulses, or pressure waves, can propagate through the network via multiple routes. The network becomes resilient, tolerant of damage, capable of maintaining function even when individual hyphae are lost. And here's something even more remarkable. Disconnected fragments can reconnect. If a portion of mycelium is physically severed from the main network, perhaps by a burrowing animal, perhaps by frost heave that shifts soil layers, those disconnected fragments don't simply die. They continue growing, if conditions permit. And if their hyphae encounter hyphae from the main network again, or if they encounter each other, they can fuse through anastomosis, reintegrating into the connected network. What was broken can be made whole again. What was separated can rejoin. This gives mycelium a kind of physical memory, or perhaps more accurately, a physical persistence. The network has no central point of failure because it has no center at all. It's distributed, redundant, self-healing. Break it apart and the pieces can find each other and reconnect. This is one reason why mycelial networks can be so ancient and so extensive. They don't die easily because they don't have vulnerable critical structures whose loss would be fatal. As anastomosis occurs repeatedly throughout the growth of a mycelial colony, something interesting emerges at the network level. Networks become statistically isotropic. Isotropic means uniform in all directions, lacking any preferred orientation. Early in a colony's growth, when it's just beginning to spread from a germinating spore, the network might have directionality, more growth in one direction than another based on where nutrients were found or how the initial hyphae happened to orient. But as the network matures, as anastomosis creates more and more interconnections, as the mycelium fills the available space more thoroughly, the network architecture becomes increasingly isotropic. This means that if you were to examine the network from any direction, at any angle, you'd see roughly the same statistical properties, the same average density of hyphae, the same frequency of branching, the same degree of interconnection. It's a kind of spatial homogeneity that emerges from the growth process itself. The mycelium doesn't deliberately create this uniform structure. There's no blueprint, no plan. But the combination of branching, anastomosis, and response to environmental factors naturally leads to networks that fill space efficiently and uniformly. This isotropy has functional advantages. It means the mycelium explores its environment comprehensively rather than leaving unexplored gaps. It means nutrients can be transported efficiently in any direction as needed. It means the network has no weak directions, no orientations where damage would be particularly catastrophic. It's robust architecture emerging from simple, repeated processes. When scientists study these networks in detail, measuring thousands of individual hyphae, analyzing branch points and fusion points, mapping the three-dimensional structure, they find consistent patterns. One such pattern involves the thickness of the hyphal strands that make up the network. While individual hyphae have the diameters we've discussed, ranging from submicron to a few micrometers, when you average across entire networks, taking into account single hyphae, bundled hyphae, and thick hyphal cords, a characteristic emerges. The average network diameter is approximately 1.3 micrometers. This is a statistical measure, of course. Some hyphae are thinner, some thicker. Some regions of the network have fine, exploratory hyphae. Other regions have thick cords formed by many hyphae running parallel, bundled together for efficient long-distance transport. But averaged across the network, taking all of these variations into account, 1.3 micrometers emerges as a typical value. It's been observed across different fungal species, in different environments, in networks of different ages. Why this particular value? It likely represents an optimization, a balance between competing demands. Thinner hyphae have more surface area relative to volume, which is advantageous for absorbing nutrients. They can also penetrate finer spaces and require less material to construct. But they're more fragile, more susceptible to damage, and less efficient at transporting materials over long distances. Thicker hyphae are more robust and better at transport, but they're more costly to build and maintain, and they can't penetrate the finest spaces. The 1.3 micrometer average suggests that across the network as a whole, mycelium strikes a balance. Fine hyphae at the growing margins and in exploratory zones bring the average down. Thicker transport hyphae and cords in the interior of the network bring it up. The result is a network architecture that's simultaneously good at exploration, at nutrient absorption, at transport, and at structural stability. Not perfectly optimized for any single function, but well balanced across multiple functions, which is precisely what you'd expect from an organism shaped by natural selection in complex, variable environments. So here we have the architecture of resilience. Septa that provide structural support while allowing functional continuity through pores. Wurn in bodies and other sealing mechanisms that act as emergency bulkheads, containing damage before it spreads. Anastomosis that weaves separate branches into interconnected networks with multiple pathways and no single points of failure. The ability of disconnected fragments to grow and potentially reconnect, ensuring that even catastrophic fragmentation need not be fatal. The emergence of statistically isotropic networks that explore space thoroughly and transport resources efficiently in all directions. And an average diameter that balances the competing demands of penetration, absorption, transport, and structural integrity. This is how mycelium stays connected despite constant perturbation. This is how these delicate threads persist in an environment filled with mechanical hazards. Not through rigidity, not through armor, but through flexibility, redundancy, and the ability to heal and reconnect. The mycelial network is tough not because it resists damage, it can't, it's too delicate, but because it survives damage, contains it, routes around it, and ultimately recovers from it. In the dark soil, in the rotting wood, through the leaf litter in between the stones, these networks persist. They're torn and they heal. They're fragmented and they reconnect. They're punctured and they seal. Season after season, year after year, the network endures. Growing, connecting, transporting, surviving. A living architecture of remarkable resilience, built from the simplest components through the most elegant principles. This is the internal world of mycelium, structured yet fluid, divided yet connected, vulnerable yet resilient. This is how the network maintains itself, how it persists, how it thrives. Through damage control systems that act in seconds, through architectural strategies that provide redundancy, through growth processes that can repair and rebuild what's been lost. The mycelium continues, always continues, beneath our feet. Connected, resilient, enduring. There's something happening in the soil that challenges our understanding of what intelligence means. We've explored the structure of mycelium, the mechanics of its growth, the architecture of its resilience. But now we come to something that seems almost impossible. Mycelium makes decisions. It solves problems. It remembers. And it does all of this without a single neuron, without anything resembling a brain, without centralized control of any kind. This is where our journey becomes truly fascinating, where we begin to see that intelligence might be far more distributed, far more fundamental, far stranger than we ever imagined. Let's start with organization. Despite the apparent simplicity of mycelial networks, just hyphae branching and fusing, when we look closely at mature colonies, we find they're not chaotic tangles. They're not random masses of threads. Instead, mycelium forms hierarchical networks. Hierarchy implies organization, structure, different levels serving different functions. And that's exactly what we find. At the coarsest level of this hierarchy, we have what might be called the primary trunk hyphae. These are the thick, mature hyphal strands, sometimes individual hyphae that have grown particularly robust, more often bundles of multiple hyphae running parallel to each other, fused or pressed together into cord-like structures. These trunk hyphae form the main transport routes through the network, the highways along which nutrients and water and cellular components flow between distant parts of the colony. They're the backbone of the system, often radiating outward from wherever the colony originated, extending toward the actively growing margins. And these primary trunk hyphae support smaller branches. From the main trunks, secondary hyphae branch off, somewhat thinner, somewhat less reinforced, but still substantial structures. These secondary branches might extend for millimeters or centimeters before themselves branching into tertiary hyphae, thinner still, more exploratory in nature. And from these, even finer branches emerge, the thinnest exploratory hyphae that probe into new substrate, that test the environment, that search for nutrients. It's remarkably similar to how trees are organized, a thick trunk supporting major branches, which support smaller branches, which support twigs, which support the finest shoots at the growing tips. Or how river systems are organized, a major channel fed by tributaries, which are fed by smaller streams, which are fed by tiny rills and rivulets. This hierarchical pattern appears throughout nature wherever transport networks need to efficiently connect many endpoints across space. And mycelium, through nothing but simple growth rules and responses to local conditions, creates these same efficient hierarchical architectures. But unlike a tree or a river, the mycelial network is constantly reorganizing. Growth adapts dynamically to nutrient gradients. When conditions change, when new resources become available, when existing resources are depleted, when environmental factors shift, the mycelium doesn't simply continue its previous growth pattern. It responds. It adapts. It reorganizes its network architecture to suit the new conditions. Imagine a mycelial network spreading through soil. Initially, nutrients might be relatively uniformly distributed, and the mycelium grows outward fairly evenly in all directions, maintaining that isotropic character we discussed. But then, in one direction, the growing margin encounters something rich, perhaps a piece of decomposing organic matter, perhaps the root of a plant with which the fungus can form a symbiotic partnership, perhaps simply a pocket of soil particularly rich in nitrogen or phosphorus. What happens? Nutrient-rich zones cause dense branching. The hyphae that first reach this resource-rich area slow their forward growth slightly, but massively increase their branching frequency. Where there might have been one hyphal tip exploring, suddenly there are 10, 20, 50 tips, all emerging in the vicinity of the nutrient source. This proliferation of branches creates a dense network in that location, maximizing the surface area in contact with the resource, maximizing the rate of absorption. Its foraging behavior, the mycelium has found food, and it deploys more exploratory apparatus to exploit that discovery thoroughly. Meanwhile, in other directions where resources are sparse, the opposite behavior occurs. Sparse zones promote rapid exploratory growth. Rather than wasting resources producing dense networks in areas where there's little to absorb, the mycelium in these regions grows quickly but minimally, extending hyphae rapidly through the barren area in search of better conditions beyond. Branching frequency drops. The hyphae grow more linearly, covering ground quickly, spending as little time and material as possible in the unprofitable region. This is decision-making, in a sense. Not conscious deliberation, but functional decision-making nonetheless. The mycelium is allocating its resources, its energy, its cellular materials, its growth capacity, in ways that maximize returns. Invest heavily where resources are abundant. Invest minimally where they're scarce. Move quickly through bad areas to reach potentially better areas beyond them. These are sound strategies, the kinds of strategies that any foraging organism might employ. But mycelium does it without any central coordination, through purely local responses at individual hyphal tips. The process of exploring new territory reveals even more sophistication. When mycelium expands into a completely unexplored area, not all hyphae play the same role. Founder hyphae pioneer new paths. These are the first hyphae to push into virgin substrate, the scouts at the very front of the advancing colony. They grow rapidly, branch relatively sparingly, and extend long distances from the established network. These founder hyphae are taking risks, in a biological sense. They're pushing into unknown territory where conditions might be favorable or might be hostile, where nutrients might be abundant or absent. Many founder hyphae will find themselves in unfavorable locations and will cease growth or die back. But some will encounter good conditions, and when they do, they send signals back to the main network, chemical signals, changes in turgor pressure, perhaps even electrical signals propagating through the fungal tissue. And then something interesting happens. Trailing hyphae reinforce those paths. Following behind the successful founder hyphae, more hyphae grow along similar routes, converging on the successful pathways, bundling together with the original pioneers, transforming what was a single exploratory thread into a reinforced cord. This is how the hierarchical network forms dynamically. The finest exploratory hyphae that succeed in finding resources become the template for secondary and eventually primary transport hyphae. The network literally grows its own infrastructure based on which exploratory missions succeed. Paths that lead to nothing are abandoned. Paths that lead to resources are reinforced and upgraded. It's adaptive architecture, infrastructure that emerges from exploration rather than from any predetermined plan. And the sophistication doesn't end there. As the network matures, as it spreads further and exploits more distant resources, something has to give. The fungus has finite resources. There's only so much carbon, nitrogen, and phosphorus available at any moment. Maintaining the entire network at full functionality would be prohibitively expensive. So the mycelium makes another kind of decision. Mature networks recycle inner regions. The oldest parts of the colony, the areas near where the original spore germinated, the regions that have already been thoroughly exploited, these interior areas are gradually cannibalized. Hyphal walls are broken down. Cellular contents are mobilized. The resources that built those hyphae are reclaimed and transported outward to fuel growth at the expanding margins. The center of the colony becomes less dense, more ghostly, a network of thinning threads, while the periphery thrives with thick, active hyphae. This recycling is essential for continued expansion. It allows the mycelium to maintain a compact, efficient network rather than accumulating dead weight in the form of extensive inner regions that no longer serve any function. It's pruning, optimization, the reallocation of limited resources from low-value to high-value structures. And again, this happens without central control. Individual hyphae in the interior simply cease to be maintained, break down, and their constituents flow through the network to where growth is active. This process of growth, reinforcement, and recycling leads to a characteristic pattern. Mycelium grows outward in rings, not solid discs. You've probably seen this if you've ever noticed fairy rings in lawns or fields, circular or arc-shaped lines of mushrooms that appear to be expanding outward from a central point. These fruiting bodies mark the edge of an underground mycelial network that's growing outward in all directions from its origin point, maintaining an active zone of dense hyphae at the periphery while the interior becomes increasingly sparse. The ring pattern is a natural consequence of the mycelial life cycle. The center has been depleted, all easily accessible nutrients in that region have been consumed. The periphery is where fresh substrate is being encountered, where nutrients are still available, where growth is worthwhile. So the mycelium concentrates its resources there, maintaining a moving front of active growth that advances outward over time. Some fairy rings are known to be centuries old, expanding outward at a steady pace year after year, marking the slow but persistent advance of ancient mycelial networks. But here's where it gets truly remarkable. Some species remember previous nutrient locations. This was discovered through elegant experiments where mycelium was allowed to explore environments with specific patterns of nutrient availability. Then those nutrients were removed and the environment was changed. When similar conditions were reintroduced, the mycelium showed biased growth toward the locations where it had previously encountered nutrients, even though there were no chemical cues currently present at those locations to guide it. How is this possible? How does an organism without a brain, without neurons, without anything like memory as we understand it, remember where food was located? The mechanisms are still being unraveled, but they appear to involve lasting changes in the architecture and biochemistry of the hyphal network. A region that previously exploited rich resources might maintain a denser network of hyphae even after those resources are depleted, and this structural persistence could bias future growth patterns. Chemical gradients that established during the initial exploitation might persist in some form, stored in the distribution of molecules within the mycelial tissue. There's even evidence for something like habituation, a decrease in response to repeated stimuli. If mycelium encounters the same non-threatening disruption repeatedly, say, a harmless change in light or vibration, it initially responds by slowing growth, but over time it ceases to respond, as if it has learned that this stimulus is not important. And this learned non-response can persist for periods that are long relative to the timescale of hyphal growth and turnover, suggesting some kind of memory encoded in the network itself. This is memory without neurons. This is learning without a brain. It forces us to reconsider what these words mean. If memory is just the storage of information about past events in a form that influences future behavior, then mycelium has memory, structural memory, chemical memory, architectural memory embedded in the very shape and composition of its network. If learning is just the modification of behavior based on experience, then mycelium learns. It changes how it grows based on what it has previously encountered. The intelligence of mycelium, if we can call it that, is radically distributed. There's no command center, no region where decisions are made. Instead, intelligence emerges from the collective behavior of millions of hyphal tips, each responding to local conditions according to simple rules, with their collective actions coordinated through the physical and chemical connections of the network itself. It's swarm intelligence, crowd intelligence, the kind of intelligence that also appears in ant colonies, in flocks of birds, in schools of fish, complex, adaptive behavior arising from simple units following simple rules. And perhaps this distributed form of intelligence is more ancient, more fundamental than the centralized intelligence of brains. After all, fungi evolved hundreds of millions of years before the first animal brains. The strategies mycelium uses to explore environments, to allocate resources, to optimize network architecture, these strategies have been refined over unimaginable spans of evolutionary time. They work. They work so well that fungi have colonized virtually every terrestrial environment on Earth, from the coldest tundra to the hottest deserts, from the poorest soils to the richest, from sea level to high mountains. What we're witnessing in the growth of mycelium is ancient wisdom, not conscious wisdom, but evolutionary wisdom, solutions to problems encoded not in neural circuits but in growth algorithms, in chemical response pathways, in the physics of branching networks. The mycelium solves mazes, not by thinking through them, but by growing through them, with unsuccessful paths dying back and successful paths being reinforced. It optimizes transport networks without calculus or computer models, simply by following local rules about where to grow thick and where to grow thin. It remembers where food was found, not in memory cells, but in the persistent structure of its own body. It makes predictions about where to search, not through reasoning, but through biased growth based on past success. It communicates within itself, not through nerve impulses, but through chemical signals, pressure waves, and the physical continuity of its cytoplasm flowing through interconnected tubes. This is intelligence without a brain. This is cognition distributed across a living network. This is problem-solving embedded in the very process of growth itself. And it's happening right now, beneath every footstep, throughout every forest, every field, every garden. Ancient networks making decisions, remembering, adapting, optimizing, all in profound silence, all in near total invisibility. The mycelium is thinking, in its own way. Not the way we think, not with words or images or conscious deliberation, but thinking nonetheless, in the language of growth and chemistry and physical structure. The forest floor is alive with cognition, with distributed intelligence making countless tiny decisions that aggregate into the coherent, adaptive behavior of the mycelial network. This is where our understanding of intelligence must expand to encompass forms of cognition entirely unlike our own. This is where we must recognize that the human brain, for all its remarkable capacities, is not the only way the universe has discovered to process information, solve problems, remember, and adapt. The mycelium has been solving these problems for hundreds of millions of years, using networks instead of neurons, chemistry instead of electricity, structure instead of circuitry. And it works. It works beautifully. Intelligence without a brain. Memory without neurons. Wisdom grown into the very fabric of a living network. The soil thinks. The forest floor remembers. and we're only beginning to understand how. We've been speaking of mycelium as threads, as networks, as filaments spreading through substrate. And this is true. But it's not the complete picture. Because mycelium, despite its apparent simplicity, despite being built from nothing but hyphae, can organize itself into specialized structures of remarkable sophistication. These aren't organs in the traditional sense. There's no mycelial heart, no fungal liver. But there are structures, differentiated tissues, specialized forms that serve specific functions. The mycelium has built-in tools, adaptations shaped by hundreds of millions of years of evolution to meet the challenges of survival in diverse and demanding environments. Let's begin with one of the most visually striking of these structures. In certain fungi, particularly those that decompose wood or need to transport resources across significant distances, the mycelium organizes itself into thick, rope-like strands that can be several millimeters in diameter and meters in length. These are called rhizomorphs, and they form rope-like nutrient highways through the substrate. The name itself is revealing. Rhizomorph means root-shaped, and indeed, when you encounter these structures in rotting wood or beneath bark, they do look remarkably like plant roots, dark and cord-like, branching and rebranching through the substrate. But they're not roots. They're bundles of hyphae, thousands or even millions of individual threads organized parallel to each other, running in the same direction, sometimes fused together, sometimes merely pressed tightly against each other, creating a macroscopic structure from microscopic components. Think of how individual fibers can be twisted together to form rope, and how rope becomes much stronger than any individual fiber could be. Rhizomorphs employ a similar principle, but they're living ropes, capable of growth, transport, and response. Within these rhizomorph structures, there's organization, specialization of function. The hyphae are not all identical. Some have differentiated into what we might call central vessel hyphae. These central vessel hyphae transport resources efficiently over long distances. They're wider than typical hyphae, with fewer crosswalls or more open septal pores, allowing for rapid, low-resistance flow of nutrients and water along the length of the rhizomorph. They function as vascular tissue, as plumbing, moving resources from sites of absorption to sites of utilization, from a rich patch of decomposing matter to a distant growing front, or from extensive absorptive networks to developing fruiting bodies. Surrounding these central vessel hyphae are supporting hyphae, often with thicker walls, providing structural integrity to the cord. There may also be surface hyphae with specialized properties, perhaps secreting protective compounds or enzymes. The rhizomorph is thus a differentiated structure, a multicellular organization with division of labor, even though it's built entirely from a single cell type, the hypha, modified in various ways for different functions. These rhizomorphs allow fungi to do something remarkable. They can span gaps. A rhizomorph can grow across a stretch of nutrient-poor soil or even through air, connecting one resource-rich location to another. In forests, certain fungi use rhizomorphs to connect multiple trees, moving nutrients between them, creating living bridges that can extend for meters beneath the forest floor. The rhizomorph is a lifeline, a supply route, allowing the fungus to access resources far from where it's actively absorbing them, to integrate distant parts of its network into a functional whole. But rhizomorphs are ephemeral structures, existing to serve the needs of active growth and transport. What happens when conditions turn harsh? When winter comes, or drought descends, or substrate is exhausted? Mycelium needs a way to survive periods when growth is impossible, when the environment turns hostile. And for this, many fungi produce another specialized structure. Sclerotia act as long-term survival structures. A sclerotium, plural sclerotia, is a hardened mass of mycelium, a compact, often dark-colored nodule that forms within or on the surface of colonized substrate. If you've ever seen fungal growth on old food in the back of the refrigerator, those hard black specks might have been sclerotia. But they're not simply dead tissue. They're highly organized survival capsules, biological time capsules designed to endure. The formation of a sclerotium begins with hyphae in a particular region stopping their exploratory growth and instead growing inward, interweaving tightly, packing together into an increasingly dense mass. The outer layers of hyphae develop thick, melanized walls, melanin providing protection against UV radiation, desiccation, temperature extremes, and enzymatic attack from other organisms. The inner hyphae store concentrated nutrients, lipids, proteins, carbohydrates, reserves that will fuel future growth when conditions improve. The entire structure becomes dormant, metabolic activity dropping to minimal maintenance levels. In this state, sclerotia can survive for months or even years. They can endure freezing, drying, being buried, being eaten and passed through an animal's digestive system. They're resistant to decay, to attack by bacteria and other fungi, to chemical stresses. They simply wait, patient as stone, for the right conditions to return. And when favorable conditions do return, when moisture and temperature are suitable, when the sclerotium is in contact with substrate that can be colonized, it germinates. Hyphae emerge from the hardened surface and begin growing outward, rapidly establishing a new mycelial network, drawing on the nutrient reserves stored within the sclerotium to fuel this initial burst of growth. What was a dormant survival structure becomes the seed of a thriving colony. It's a strategy parallel to seed formation in plants: concentrate resources, harden defenses, wait out bad times, resume growth when opportunity returns. Not all specialized mycelial structures are about transport or survival. Some are about creating specific textures or forms needed for particular functions. Consider the tissue that forms the flesh of many mushrooms, or the leathery crusts that some fungi form on wood surfaces. These tissues have names, classifications, structures that merit closer attention. One type is called prosenchyma. Prosenchyma forms felt-like tissues. In prosenchyma, the hyphae remain recognizable as individual threads, but they're woven together in a dense, interlocking mat. Imagine felt fabric, where individual fibers are tangled and compressed but still distinct. That's prosenchyma, hyphae running in various directions, overlapping, branching through each other's spaces, creating a cohesive tissue that has structural integrity but retains a fibrous character. When you tear a mushroom cap and see a stringy, fibrous texture, you're seeing prosenchymatous tissue. The individual hyphae can still be distinguished, still retain their tubular structure, but collectively they create something more than just a loose network. They create tissue, fabric, structure. But fungi can go further in tissue differentiation. Some create tissues where the hyphal nature is almost entirely obscured, where the structure looks remarkably similar to the cellular tissues of plants. This is called pseudoparenchyma, and pseudoparenchyma resembles plant cells. The prefix pseudo is important. It means false or resembling. This isn't true parenchyma as found in plants, where individual cells divide to create tissue. Instead, it's hyphae that have become so swollen, so tightly packed, so modified in form that they appear as rounded or polygonal cells pressed against each other, virtually indistinguishable from plant tissue without careful microscopic examination. Pseudoparenchymatous tissue forms the firm, fleshy context of many mushrooms, the solid, homogeneous flesh that doesn't appear fibrous when broken. The hyphae have swelled, shortened, lost their obviously tubular character, and fit together like cells in an onion or apple. This allows for dense, strong tissues that can support substantial structures like large mushroom caps. It's another example of how limited building blocks, just hyphae, can be organized in different ways to create dramatically different properties and appearances. Between the extremes of loose mycelial networks and compact sclerotia, there's another level of organization particularly important for resource transport. Mycelial cords organize transport cores within their structure. These cords are intermediate between ordinary mycelium and full rhizomorphs, thicker than individual hyphae, organized for transport, but less rigidly structured than true rhizomorphs. Within a mycelial cord, you'll find differentiated zones, a central core of parallel hyphae optimized for rapid transport, sometimes with particularly wide diameter or sparse septa, surrounded by a cortex of more typical hyphae providing structure and perhaps absorptive capacity. These cords form the intermediate highways of mycelial networks, not as massive as rhizomorphs, but more organized than loose mycelium, creating the branching pattern of major and minor routes that efficiently distribute resources throughout the colony. They can form, be reinforced when useful, or be degraded and reabsorbed when no longer needed. Their dynamic infrastructure, growing and shrinking in response to the network's current needs. All of these structures we've discussed so far develop within substrate, in soil, in wood, in organic matter. But some fungi need to escape the substrate, to send structures into the air. Perhaps to position spore-bearing structures where wind can disperse the spores. Perhaps to attach to passing insects. Perhaps simply to escape the moisture of saturated substrate where bacterial and protozoan predators are too abundant. For this, fungi develop aerial hyphae, and these require special adaptations. Water is a problem in air. Specifically, water loss. A hypha projecting into air is exposed to evaporation, and the thin-walled, moisture-filled tubes we've been describing would rapidly desiccate, their turgor pressure would drop, growth would cease, the hyphae would collapse and die. But many fungi solve this problem elegantly. Aerial hyphae develop hydrophobic coatings on their surfaces. Hydrophobic, water-repelling. These coatings are typically composed of special proteins called hydrophobins, and hydrophobins protect surfaces from moisture loss and from wetting. This might seem contradictory. How does a water-repelling coating prevent water loss? The answer lies in the coating's properties. Hydrophobins self-assemble into amphipathic layers on the hyphal surface, meaning one side of the layer interacts with the water-containing hyphal wall, while the other side, facing the air, is intensely hydrophobic. This hydrophobic outer surface has multiple effects. It dramatically reduces the rate at which water can evaporate from the hyphal wall, much as a layer of oil on water reduces evaporation. It prevents water from condensing on the hyphal surface, which is important because surface water films would make the hyphae vulnerable to bacterial colonization and would interfere with spore dispersal. And it reduces the surface tension that would otherwise cause aerial hyphae to collapse together when they touch, allowing them to maintain separate, spreading growth into air. Hydrophobins are remarkable proteins, among the most surface-active biological molecules known, meaning they're extraordinarily good at concentrating at interfaces between water and air or water and hydrophobic surfaces. They spontaneously assemble into highly stable layers just one protein molecule thick, creating an effective barrier with minimal material investment. They're so effective that biotechnology researchers are exploring their use in everything from medical coatings to food science applications. But in mycelium, they serve their ancient function, protecting aerial hyphae, enabling the fungus to escape the confines of its substrate and project structures into the air. These hydrophobin-coated aerial hyphae are what gives some fungal growths their fuzzy or powdery appearance. The coating scatters light in a way that makes the hyphae appear white or gray, and the water-repelling nature means these aerial structures don't mat down or clump when moistened. They maintain their airy, separated structure. Touch a patch of mold on bread and feel how it's dry and powdery despite the moisture in the bread itself. That's hydrophobins at work, waterproofing the aerial mycelium. So here we have the toolkit, the specialized structures that transform simple hyphal networks into a versatile, adaptable organism capable of thriving in diverse conditions. Rhizomorphs that transport resources across distances, with their specialized vessel hyphae forming living pipelines. Sclerotia that allow survival through hard times, dormant and hardened, waiting months or years for favorable conditions to return. Prosenchyma creating fibrous tissues, felt-like inflexible. Pseudoparenchyma forming dense, fleshy structures that mimic plant tissues. Mycelial cords with organized transport cores, forming the highways and byways of the network. Aerial hyphae escaping into the air, protected by hydrophobin coatings that repel water and prevent collapse. All of this from hyphae. All of this complexity, all this functional diversity, built from variations on a single basic structure, the tubular filament. Modified in diameter, in wall thickness, in branching pattern, in packing arrangement, in surface chemistry, but fundamentally just hyphae, organized in different ways to meet different needs. It's like the entire organism is written in a very simple language, the language of hyphal growth, but through syntax, through organization, through the arrangement and modification of these simple units, complex and sophisticated structures emerge. A rope-like rhizomorph is a sentence about long-distance transport. A sclerotium is a paragraph about survival. A sheet of pseudoparenchymatous tissue is a chapter about structure and support. And the entire organism, the entire mycelial colony with all its varied structures, is a book written in this simple but infinitely flexible language. These structures aren't planned. There's no blueprint, no architect. They emerge from the growth process itself, from hyphae responding to conditions, from local interactions giving rise to organized complexity. And yet they work. They work so well that fungi have been employing these strategies for hundreds of millions of years, in environments from the Arctic to the tropics, in habitats from pristine forests to the interiors of buildings, from rich soils to bare rock faces where almost nothing else can grow. The mycelium adapts. It builds what it needs from what it has. And what it has are hyphae, simple tubes that can be organized into rhizomorphs or sclerotia, into prosenchyma or pseudoparenchyma, into cords or aerial filaments, into any of 1,000 variations that serve the organism's needs in its particular environment at its particular moment. This is the engineering of life at the fungal scale. Not engineering with gears and levers, not engineering with circuits and codes, but engineering with growth, with self-assembly, with structures that build themselves according to simple rules that produce complex results. Built-in tools, specialized structures, all grown from the same fundamental building block, all emerging from the same basic process of hyphal extension and branching. The mycelium carries its toolkit within itself, written in its DNA, expressed through its growth, manifested in these remarkable structures. Rope-like highways, survival capsules, felt and flesh, transport cores, waterproof coatings. All the tools needed to survive, to grow, to persist, to thrive. All of it built from threads. All of it grown in darkness. All of it working, right now, beneath the surface, in the hidden spaces, in the world we walk over but rarely think about. The mycelium, with its specialized structures, its built-in tools, its elegant solutions to the challenges of existence. Growing, adapting, surviving, century after century, in silence and in darkness, beneath our feet. There is a conversation happening beneath the forest floor. Not in words, not in sounds, but in molecules and minerals, in carbon and chemistry. Trees are speaking to fungi. Fungi are speaking to trees. And through the mycelial network, trees are speaking to each other, sharing resources, sending warnings, supporting their offspring. This is perhaps the most profound role that mycelium plays in terrestrial ecosystems, not as a decomposer, not as a solitary organism, but as a connector, a mediator, a living bridge between the plant world and the mineral soil. This is symbiosis at its most elegant. This is the underground internet. Let's begin with the fundamental relationship. Mycelium forms mycorrhizal relationships with the roots of most land plants. Mycorrhiza, the word itself tells the story. Myco means fungus, rhiza means root. Fungus root. It's a partnership that's so ancient, so fundamental, that some paleobotanists believe plants could not have colonized land without it. The earliest land plants, lacking true roots, may have relied entirely on fungal partners to gather water and minerals from the substrate. Even today, it's estimated that over 90% of plant species form mycorrhizal associations. This isn't a rare adaptation, an unusual strategy. This is the norm. This is how most plants interface with the soil. The fungal partner, the mycelium, gains something essential from this relationship. It gains carbon. Fungi cannot photosynthesize. They cannot capture the energy of sunlight and convert it into sugars the way plants can. But plants are extraordinarily good at this, capturing light energy and fixing carbon from the air into organic compounds. Some of these compounds, sugars, lipids, amino acids, flow from the plant through its roots to the fungal partner. It's payment, essentially, for services rendered. The plant feeds the fungus. And what service does the fungus provide? It provides access. Access to water and minerals that the plant's roots alone could never efficiently obtain. Here's why this matters so much. Plant roots are relatively thick structures, often a millimeter or more in diameter. They can only explore the soil to the extent that they can physically penetrate it, and they can only absorb nutrients that are within a very short distance of their surface, typically just a millimeter or two. Much of the soil's volume is simply inaccessible to roots, and nutrients that are more than a few millimeters away from a root surface might as well be on another planet for all the good they do the plant. But mycorrhizal hyphae? They're microscopic. They can penetrate the tiniest soil pores. They can extend far beyond the root zone. And they do. Extra-radical hyphae extend far into soil, sometimes tens of centimeters away from the root they're associated with. These are the hyphae that grow outward from the root surface into the surrounding soil, exploring, absorbing, accessing resources in a volume of soil orders of magnitude larger than the roots could reach alone. A single mycorrhizal fungus associated with a plant's roots might extend hyphae into hundreds of cubic centimeters of soil, creating an absorptive surface area that dwarfs the surface area of the roots themselves. These extra-radical hyphae are absorbing water, yes, but more importantly they're absorbing mineral nutrients, nitrogen, phosphorus, potassium, calcium, micronutrients like iron and zinc. Phosphorus is particularly important. Phosphorus in soil exists largely in forms that are insoluble, bound to soil particles, immobile. It doesn't flow to roots with water. Roots can only access the phosphorus immediately around them, and once they've depleted that local supply, they're stuck unless they grow new roots into fresh soil. But mycorrhizal hyphae, with their enormous surface area and their ability to extend far beyond the root zone, can access much larger phosphorus pools. They can even produce acids and enzymes that liberate phosphorus from insoluble compounds, making it available where it otherwise wouldn't be. But how do these minerals get from the hyphae in the distant soil to the plant? This is where the other half of the mycorrhizal structure comes in. While extra-radical hyphae extend outward into soil, other hyphae, intra-radical hyphae, exist within or in intimate contact with the root tissue itself. These intra-radical hyphae exchange carbon and minerals at the interface between fungus and plant. The plant exports sugars and other carbon compounds into the root tissue, and the hyphae absorb them. Simultaneously, the hyphae, which have transported minerals inward from their extra-radical portions through the mycelial network, transfer these minerals to the plant. It's a trade, a barter system operating at the cellular level. The precise architecture of this exchange depends on the type of mycorrhiza. There are several distinct forms, evolved independently in different plant and fungal lineages, each with its own structural characteristics. One major type is ectomycorrhizae. These form primarily with trees, oaks, pines, birches, beeches, and the relationship has distinctive features. Ectomycorrhizae form a fungal mantle on roots, a sheath of fungal tissue that completely envelopes the finest root tips. If you've ever gently excavated the roots of a pine tree and noticed that the finest root tips look slightly swollen and are covered in what appears to be a fuzzy or cottony coating, you've seen an ectomycorrhiza. That coating is the mantle, a dense layer of fungal hyphae, often several cell layers thick, completely surrounding the root. But the fungus doesn't just coat the surface. It penetrates between the cells of the root's outer layers. Hard nets weave between root cells, creating an intricate interface where fungal hyphae snake through the spaces between plant cells without actually penetrating into the cells themselves. Imagine the root's outer tissue as a brick wall, and the hard net as mortar that's somehow alive, fungal hyphae filling the gaps between cells, maximizing surface contact. This is where exchange occurs, minerals flowing from hyphal cytoplasm into the apoplast, the space between plant cells, from where the plant can absorb them. Carbon compounds flowing in the reverse direction, from plant cells into the intercellular spaces where hyphae take them up. The other major type of mycorrhiza is endomycorrhiza, specifically arbuscular mycorrhizae, which form with most herbaceous plants, many trees, and most agricultural crops. These have a different architecture. Here, the fungus doesn't form an extensive external mantle. Instead, fungal hyphae actually penetrate into the root cortex cells themselves. But they don't break the plant cell membranes. Instead, they push inward, invaginating the membrane so that the fungal hypha becomes surrounded by plant membrane but remains technically outside the plant cytoplasm. And inside these penetrated cells, the fungus creates remarkable structures. Arbuscules form tree-like exchange structures within the plant cells. An arbuscule is a highly branched hyphal structure that looks, under the microscope, like a tiny tree. The fungal hypha enters the plant cell and then branches repeatedly, dichotomously, creating finer and finer branches, until the entire structure resembles a miniature deciduous tree in winter. These repeated branchings create an enormous surface area for exchange. The arbuscule might increase the interface area between fungus and plant by a factor of 10 compared to a simple unbranched hypha. This is where the trade happens in arbuscular mycorrhizae. The plant cell, with an arbuscule growing inside it, becomes a specialized exchange site. Carbon flows from the plant across the membrane surrounding the arbuscule, feeding the fungus. Phosphorus and other nutrients flow from the fungal tissue across that same membrane into the plant cell. The arbuscule is ephemeral. It typically lasts only days before collapsing and being digested, with new arbuscules forming continuously in other cells. It's a dynamic interface, constantly being rebuilt, maintaining the exchange that both partners depend on. These mycorrhizal relationships, whether ecto or arbuscular, create something profound at the ecosystem level. Because a single fungal mycelium doesn't associate with just one plant. A single mycelial network can extend from the roots of one tree to the roots of another, and another, and another. Common mycorrhizal networks link multiple plants, sometimes many plants of different species, all connected through the same fungal mycelium. This has been demonstrated repeatedly through isotope tracing studies where researchers label the carbon in one plant's photosynthesis and then detect that labeled carbon appearing in other, distant plants, proof that they're connected, that resources are moving between them through the mycelial network. These networks have been called the Wood Wide Web, and while that's perhaps a bit too anthropomorphic, it captures something true. The mycelium is connecting the forest in ways that fundamentally change how we must think about forests. A forest is not a collection of individual trees competing for resources. It's a network, a superorganism perhaps, where trees are nodes and mycelial connections are the links, and resources, carbon, water, nitrogen, phosphorus, even possibly defensive compounds and signaling molecules, flow through the network according to needs and availabilities that we're only beginning to understand. One of the most striking findings involves resource transfer between trees of different ages and conditions. Carbon can flow from mature trees to seedlings through the mycelial network. Large, well-established trees with deep roots and extensive canopies are photosynthetic powerhouses, producing far more carbon than they need for their own metabolism. Meanwhile, seedlings on the forest floor, shaded by the canopy, struggling to photosynthesize in the low light, are carbon-starved. But if both are connected to the same mycorrhizal network, carbon can flow from the mature tree through the mycelium to the seedling, subsidizing its growth during the vulnerable early years when it's establishing itself. This isn't altruism. The fungus is moving carbon around its network according to its own needs and the source-sink dynamics of the system, but the effect is that mature trees support their offspring and the offspring of other trees, that the forest becomes more stable, that regeneration is facilitated. Some researchers have found that survival rates of seedlings connected to mycorrhizal networks are significantly higher than those of seedlings that are not connected. The network is providing a subsidy, a boost, helping young trees establish in an environment that would otherwise be too resource poor for survival. But how does this relationship even begin? How do plant roots and fungal hyphae find each other in the vast, dark complexity of the soil? The answer involves chemical signaling of extraordinary specificity. Mycelium detects plant strigolactones. Strigolactones are a class of plant hormones that roots release into the soil, among other things, as signals to potential mycorrhizal partners. They're saying, in chemical language, "I'm here, I'm a root, I need a fungal partner." Mycorrhizal fungi have receptors that detect these strigolactones even at incredibly low concentrations. When a hypha detects strigolactone molecules, it responds by growing toward higher concentrations, following the chemical gradient toward its source, the root. The strigolactone also triggers changes in the fungus's gene expression, activating the genes needed for symbiosis, preparing the mycelium for the complex developmental changes required to form the mycorrhizal structure. It's a molecular conversation. The plant speaks first, releasing strigolactones. The fungus hears and responds, growing toward the signal. As the hypha approaches the root, other signals are exchanged. The root may release flavonoids or other compounds that further modulate fungal growth and development. The fungus releases its own signals, chitin fragments, proteins, small RNAs, that tell the plant, "I'm approaching. Prepare for symbiosis. Don't treat me as a pathogen." This is crucial because plants have sophisticated immune systems designed to detect and repel invading microorganisms. The mycorrhizal fungus must, in effect, negotiate a truce, convince the plant to allow its penetration and colonization. When both partners are communicating correctly, when the signals align, symbiosis proceeds. The root allows hyphal penetration. The fungus forms the appropriate structures, mantle and heart net, or arbuscules, depending on the type of mycorrhiza. Exchange begins. Carbon flows one direction, minerals flow the other. Both partners benefit. The relationship stabilizes. And then, through the mycelium connecting multiple plants, those plants become linked. Not physically in the sense of being grafted together, but functionally linked through the flow of resources and possibly information through the fungal intermediary. A mature tree's surplus carbon can reach a distant seedling. A plant under insect attack might, through the mycelial network, signal other plants to upregulate their defensive chemistry. A tree dying of drought might reallocate its remaining resources through the network to its neighbors. These possibilities are still being researched, still being debated, but the basic reality is clear. The mycelial network connects plants in ways that make the forest more than the sum of its individual trees. This is symbiosis at the ecosystem scale. This is cooperation, though not conscious cooperation, between kingdoms of life. Plant and fungus, autotroph and heterotroph, photosynthesizer and decomposer, brought together in a relationship so ancient that it may predate the evolution of true roots themselves, so fundamental that most terrestrial ecosystems would collapse without it. The mycelium has become essential to plant life, and plant life has become essential to the mycelium. Neither can thrive without the other in most natural settings. The underground internet, the Wood Wide Web, the mycorrhizal network. Whatever name we give it, it represents one of the most important interfaces in terrestrial ecology, the interface between plants and soil, mediated by fungi, enabling the flow of carbon from atmosphere to earth and the flow of minerals from earth to plant. Without this network, forests as we know them could not exist. Grasslands could not exist. Agriculture, in its current form, would be impossible. And it's all happening through mycelium. Through those microscopic threads we've been discussing, extending from root to root, absorbing from vast volumes of soil, transporting across distances, exchanging at cellular interfaces, connecting the forest into a living, communicating network. Extra-radical hyphae reaching far into soil, intraradical hyphae intimately entwined with root tissue, mantles and hardig nets and arbuscules creating the architecture of exchange. Common networks linking dozens or hundreds of plants into functional communities. The conversation continues, beneath our feet, in the darkness. Plants speaking in stridolactones. Fungi responding with growth and recognition. Carbon flowing one way, minerals flowing the other. Mature trees supporting seedlings. The network redistributing resources according to need and availability. All of it mediated by mycelium, that ancient network, that living bridge between kingdoms, that essential partner without which terrestrial life as we know it would be impossible. The underground internet. Always on. Always connected. Always facilitating the conversation between plant and soil, between individual and community, between the world of light above and the world of minerals below. The mycelium, connecting everything, supporting everything, essential to everything. This is symbiosis. This is cooperation. This is the partnership that made forests possible, that sustains them still, that works every moment of every day in silence and darkness, beneath the surface, in the hidden world. The mycelium and the root, the fungus and the plant, together as they have been for hundreds of millions of years. Talking, trading, connecting, sustaining. The conversation continues. In the hidden world beneath the surface, the mycelium does not exist in isolation. The soil is crowded, competitive, often hostile. Bacteria by the billions. Protozoa hunting through water films. Nematodes, microscopic worms, grazing on hyphal tissue. Insects with chewing mouthparts. Other fungi, competing for the same resources. And above, through the mycorrhizal connections, the plants face their own threats, herbivorous insects, pathogenic microbes, drought, damage. The mycelium must defend itself. It must communicate danger. It must fight, chemically and strategically, for survival. This is chemical warfare at the microscopic scale. These are warning systems operating through living networks. This is the mycelium as sentinel, as defender, as chemical weapons laboratory. Let's begin with communication itself, because defense often starts with information. We've discussed how mycelium can respond to local conditions, how individual hyphal tips sense and react to their environment. But information can also travel through the network, allowing distant parts of the mycelium to coordinate responses. Mycelium transmits electrical and chemical signals across its network, enabling communication that spans surprising distances. The electrical signals are perhaps most remarkable. Researchers have detected waves of electrical potential changes propagating through mycelial networks, traveling along hyphae at speeds of up to a few centimeters per minute. These aren't nerve impulses. There are no neurons here, no voltage-gated ion channels of the type that animals use. Instead, they appear to be waves of depolarization and repolarization traveling through the cytoplasm and along cell membranes, possibly mediated by calcium ions or other charged molecules flowing through the hyphal network. When one part of a mycelium is disturbed, say, by physical damage or exposure to a chemical irritant, electrical signals propagate outward from that location, potentially alerting the rest of the network to the disturbance. The function of these electrical signals is still being unraveled. They might coordinate growth responses, trigger the production of defensive compounds in regions distant from the initial threat, or modulate the transport of resources within the network. They represent a form of rapid, network-wide communication that allows the mycelium to respond as an integrated whole rather than as a collection of independent parts. Chemical signals travel more slowly but carry more specific information. A molecule produced in one part of the network can diffuse through the streaming cytoplasm, flowing through hyphal interiors and passing through septal pores, eventually reaching distant locations. These molecules might be hormones that regulate development, defensive compounds that prepare the network for attack, or signals that indicate resource abundance or scarcity. The mycelial network becomes a chemical messaging system, with information encoded in molecular form flowing through the living cables of the fungal tissue. And these signals don't stop at the boundaries of the fungus. Through mycorrhizal connections, the mycelium can communicate with plants, and perhaps more remarkably, it can allow plants to communicate with each other. Networks warn plants of pests through the mycelial connections. This has been demonstrated in elegant experiments. When one plant is attacked by herbivorous insects or pathogenic fungi, that plant's defensive chemistry changes. It upregulates genes for toxins, for compounds that make its tissues less palatable or less nutritious, for volatile molecules that attract predators of the herbivores. This is a standard plant defense response. But here's what's remarkable. Neighboring plants connected to the same mycorrhizal network, even if they're not directly experiencing any attack themselves, also begin to upregulate their defensive chemistry. Something is traveling through the network, chemical signals, perhaps, or even RNA molecules that can influence gene expression in the recipient plant. The mycelium is carrying a warning. Danger detected. Herbivores active in this area. Prepare your defenses. Plants that receive this warning and preemptively strengthen their defenses suffer less damage when herbivores eventually reach them compared to plants that aren't connected to the network and must wait until they're actually attacked to begin responding. This transforms how we must think about plant communication. Plants aren't isolated individuals. Through the mycelial network, they're participants in a neighborhood watch system, able to receive early warning of threats and prepare accordingly. The mycelium, serving as the communication infrastructure, benefits as well. Healthier plants mean more carbon supply for the fungal partner, so facilitating plant defense is in the fungus's interest. But communication and warning are only part of the story. The mycelium must also feed itself, must break down the complex materials in its environment to access the nutrients locked within them. And it must defend itself directly against the myriad organisms that would consume it or compete with it. Let's consider feeding first, because the chemistry of feeding is also part of the chemistry of offense. Mycelium cannot engulf food particles the way an amoeba might. It cannot chew, cannot mechanically break down materials. Instead, it must digest externally. Enzymes digest polymers externally, outside the hyphal cells. The mycelium secretes powerful enzymes, cellulases that break down cellulose, ligninases that attack lignin, proteases that cleave proteins, lipases that split fats, into the surrounding substrate. These enzymes work outside the fungal cells, breaking the large, complex polymer molecules into smaller units. Cellulose, for instance, is a polymer of glucose, thousands of glucose molecules linked together in long chains. A cellulase enzyme breaks the bonds between glucose units, liberating them. Lignin, the complex aromatic polymer that makes wood hard and resistant to decay, is attacked by specialized oxidative enzymes that break its intricate structure down into smaller aromatic compounds. Proteins are cleaved into amino acids. Lipids are split into fatty acids and glycerol. Once broken down into these smaller molecular units, monomers, they can be absorbed. Monomers are absorbed by diffusion across the hyphal cell membrane. Glucose molecules, amino acids, fatty acids, mineral ions, all small enough now to pass through transport proteins in the fungal cell membrane, moving from the external environment into the cytoplasm where they can be used for energy, for building new cellular structures, for all the metabolic needs of the growing mycelium. But this strategy, secreting enzymes and absorbing the breakdown products, has a vulnerability. The same small molecules that the mycelium is absorbing can also be absorbed by bacteria, by other fungi, by any organism in the vicinity. The mycelium is, in effect, creating a resource bonanza in its immediate surroundings, and competitors will move in to exploit it. This is where the warfare begins. The mycelium defends its digestive zone chemically. Antibacterial peptides like copsin are secreted into the substrate. Copsin and similar compounds are small proteins that can insert into bacterial cell membranes, creating pores, disrupting membrane integrity, ultimately killing the bacteria. They're selective. They target bacterial cells but don't harm the fungal cells that produce them. By secreting these antibacterial compounds in the same zone where digestive enzymes are active, the mycelium creates an exclusion zone, a territory where it can safely absorb nutrients without facing overwhelming bacterial competition. Different fungi produce different antibacterial compounds, a vast chemical arsenal refined by hundreds of millions of years of evolutionary arms races. Some produce simple organic acids that lower pH to levels bacteria cannot tolerate. Others produce complex alkaloids or terpenes that interfere with bacterial metabolism. And some, famously, produce antibiotics that have revolutionized human medicine. Penicillin forms during microbial competition. The classic example, of course, is Penicillium fungi, bread molds and their relatives, which produce penicillin when growing in competition with bacteria. Penicillin interferes with bacterial cell wall synthesis, causing bacterial cells to burst from osmotic pressure. For the fungus, it's a defensive compound, a way to eliminate bacterial competitors from its territory. For humans, it became the first widely used antibiotic, saving countless millions of lives. But its original function, its evolutionary purpose, is warfare, chemical warfare at the microbial scale. The soil is filled with these chemical battles. Fungi producing antibacterial compounds. Bacteria producing antifungal compounds in response. Other fungi producing compounds that inhibit the first fungus. It's an endless, ancient war, fought with molecules, where survival depends on the ability to produce toxins that harm competitors while remaining immune to those toxins yourself. But bacteria aren't the only threat. Nematodes, microscopic roundworms, are abundant in soil and many species feed on fungal hyphae. They're grazing animals at the microscopic scale, chewing through hyphal tissue, consuming the cytoplasm, leaving damaged or destroyed hyphae in their wake. Some fungi have evolved remarkable defenses. Lectins defend against nematodes. Lectins are proteins that bind to specific carbohydrates, and some fungal lectins recognize and bind to molecules on the surface of nematode intestinal cells. When a nematode ingests hyphae containing these lectins, the lectins bind to the nematode's gut lining and disrupt its function, causing the nematode to stop feeding, to weaken, potentially to die. Other fungi have evolved even more dramatic anti-nematode strategies. Some form hyphal loops that constrict when touched, mechanically trapping nematodes. Others produce sticky knobs on their hyphae that nematodes adhere to, then send hyphae into the trapped nematode's body to digest it from within. The hunter becomes the hunted. These predatory fungi are relatively rare, but they demonstrate the extremes to which the evolutionary arms race between fungi and nematodes has gone. What's particularly fascinating is that fungal defenses aren't simply static, always-on systems. They can be induced, upregulated in response to attack, and, remarkably, the memory of past attacks can persist, allowing faster, stronger responses to subsequent attacks. Grazing triggers epigenetic immune memory in some fungal species. When nematodes or other grazers consume hyphal tissue, the surviving parts of the mycelium don't just heal the damage. They remember, in a molecular sense, that they've been attacked. This memory isn't encoded in changes to DNA sequence. That would be genetic memory, mutations, and selection. Instead, it's epigenetic memory, chemical modifications to the DNA or to the histone proteins around which DNA is wrapped, modifications that change which genes are expressed without changing the genes themselves. Histone modifications sustain defense responses long after the initial attack has ended. Methyl groups or acetyl groups attached to specific amino acids in histone proteins can keep defensive genes in an active state, easily and rapidly transcribed when needed. The result is that mycelium that has been grazed before responds more quickly and more strongly when grazed again. Defensive compounds are produced faster and in higher concentrations. Growth patterns change to favor forms that are less vulnerable to grazing, perhaps thicker-walled hyphae, or growth into substrates that grazers cannot easily access. The mycelium has learned, biochemically, from experience. It has immune memory, not unlike the immune memory that protects you from catching the same infection twice. This epigenetic memory can persist for days or even weeks, far longer than you'd expect if it were just a matter of proteins and enzymes lingering in the cytoplasm. The modifications to histones are stable, maintained through multiple rounds of hyphal growth and cell division, ensuring that the defensive state persists throughout the affected region of the network. Its memory encoded in chromatin structure, in the three-dimensional organization of DNA and proteins in the nucleus. But defense isn't always about aggression or toxicity. Sometimes it's about stealth, about avoiding detection. Many fungi that form arbuscular mycorrhizae need to penetrate plant roots and colonize plant tissue without triggering the plant's immune system. Plants have sophisticated pattern recognition receptors that detect molecular signatures common to microbes, chitin being a prime example. Chitin in fungal cell walls should, theoretically, trigger strong plant defenses. But arbuscular mycorrhizal fungi have found a workaround. Monon layers mask immune signals on the hyphal surface. Monons are polysaccharides, polymers of the sugar mannose, and when deposited on the hyphal surface, they cover the chitin, preventing plant receptors from detecting it. It's molecular camouflage. The fungus is still there, still penetrating the root, still forming arbuscules, but it's wearing a chemical disguise that tells the plant immune system nothing to see here, I'm not a threat. This allows the symbiosis to proceed without the plant treating its fungal partner as a pathogen. The monon masking isn't perfect. There are still some immune responses during mycorrhizal colonization, just heavily dampened ones. It's a negotiated peace, facilitated by chemical signaling and molecular disguise. The fungus downregulates its most obvious immune triggering signatures. The plant downregulates its most aggressive defensive responses. Both benefit from the partnership that results. So here we have the complete picture of mycelial communication and defense. Electrical signals flashing through the network at centimeters per minute, coordinating responses across the colony. Chemical messages flowing through cytoplasmic streams, carrying information encoded in molecular form. Warnings transmitted through mycorrhizal networks, alerting plants to approaching threats. Digestive enzymes secreted externally, breaking down polymers into absorbable monomers. Antibacterial peptides like copsin creating exclusion zones around feeding hyphae. Penicillin and other antibiotics forming during competition, chemical weapons in microscopic warfare. Lectins disrupting nematode feeding. Epigenetic immune memory, sustained by histone modifications, allowing faster responses to repeated attacks. Monon layers masking immune triggers, allowing symbiotic fungi to colonize plant tissue without triggering defensive responses. All of this is chemistry. All of it is molecular interaction, proteins and small molecules, binding and reacting, signaling and defending. But the aggregate result is behavior, is strategy, is a complex response system that allows mycelium to survive in one of the most competitive, dangerous environments on Earth, the soil, teeming with organisms that would eat the fungus or compete with it for resources. The mycelium defends itself not with armor, not with speed, not with size or strength, but with chemistry. It fights with molecules. It warns with signals. It remembers with epigenetic marks. It disguises itself with surface coatings. It's warfare at the nanoscale, communication at the molecular level, memory encoded in chromatin structure. And all of this is happening right now, constantly, in every handful of soil, in every rotting log, in every mycorrhizal association, in every forest. The chemical conversations, the warning signals, the enzymatic digestion, the antibacterial warfare, the epigenetic memory formation, all of it ongoing, all of it essential to the survival of the mycelial networks that, in turn, are essential to the ecosystems they inhabit. This is the hidden complexity of the apparently simple fungal threads spreading through substrate. The war continues. The signals propagate. The warnings are transmitted. The defenses hold. And the mycelium persists, adapting, defending, communicating, surviving through chemistry and through the ancient strategies encoded in its genome and expressed through its remarkable biochemical capabilities. The chemical warfare and warning systems of mycelium, silent, invisible, essential, elegant. Working every moment, in the darkness, beneath the surface, in the hidden world. We have been speaking of mycelium as if it were a singular entity, a pure fungal network spreading through soil and substrate. But this is a simplification, a convenient fiction that makes the story easier to tell. The truth is more complex, more intertwined, more collaborative. Mycelium never works alone. Its hyphae are colonized by bacteria. Its tissues harbor yeasts. Its metabolic processes are enhanced by actinomycetes. It partners with other fungi, shares resources with them, competes with them, sometimes all at once. Even viruses play a role, attacking some members of the community while protecting others. The mycelium is not an individual. It is an ecosystem, a community, a hybrid network of multiple organisms working together, competing, coexisting in relationships so intimate that the boundaries between self and other become blurred. Let's start inside the hyphae themselves. If we could peer into the cytoplasm of many fungal hyphae with sufficient magnification, we would find something unexpected. Bacteria. Not attacking from outside, not accidentally trapped, but living inside the hyphal tubes, dwelling within the fungal cells as permanent or semi-permanent residents. Mycelium hosts bacterial endophytes. The term endophyte means inside the plant, but here it's used more broadly to mean any organism living inside another. These bacterial endophytes exist within the hyphal cytoplasm or between the cell wall and membrane, taking up residence in the fungal tissue. Why would a fungus tolerate bacteria inside its own cells? The answer, as so often in biology, is mutual benefit. The bacteria provide services that the fungus needs. One of the most important is nitrogen fixation. Many fungal endophytes are bacteria capable of fixing atmospheric nitrogen, converting N2 gas from the air into ammonia, a form of nitrogen that can be used to build proteins, nucleic acids, and other essential biomolecules. Nitrogen is often the limiting nutrient in terrestrial ecosystems, and the ability to access atmospheric nitrogen, which is abundant but chemically inert, is tremendously valuable. The bacteria fix nitrogen using specialized enzymes called nitrogenases, supplying the fungus with ammonia or amino acids. And in return, nitrogen is exchanged for carbon. The fungus supplies the bacteria with sugars and other carbon compounds, the products of its decomposition or the carbon it receives from plant partners in mycorrhizal associations. It's a trade, carbon for nitrogen, and both partners benefit. The fungus gains access to nitrogen it couldn't otherwise obtain. The bacteria gain a protected environment inside the hyphae and a reliable carbon supply. This partnership can be so intimate that some fungal species have never been found without their bacterial endophytes, and attempts to cure the fungus of its bacteria often result in the fungus growing poorly or not at all. The two organisms have become metabolically interdependent, each relying on the other for essential nutrients. The mycelial network is not purely fungal, it's a chimera, a hybrid of fungus and bacteria working as an integrated unit. But bacteria aren't the only microbial partners. Actinomycetes enhance antibiotic production in many fungal species. Actinomycetes are a group of bacteria, often filamentous-like fungi, famous for producing a huge diversity of antibiotics. Many of the antibiotics used in human medicine, streptomycin, tetracycline, erythromycin, come from actinomycetes. When actinomycetes colonize fungal hyphae or grow in close association with mycelial networks, something interesting happens. The fungus upregulates its own antibiotic production. The actinomycetes do the same. The result is a chemical arsenal more diverse and more potent than either organism would produce alone. This makes sense from an ecological perspective. Both fungus and actinomycetes are competing with other soil microorganisms for resources. By coordinating their antibiotic production, by creating overlapping zones of chemical inhibition, they create a more effective defensive perimeter. Bacteria that might be resistant to fungal antibiotics might succumb to actinomycete antibiotics, and vice versa. Together, they secure a larger territory, a more exclusive feeding zone, than either could manage independently. It's an alliance based on shared enemies, a partnership of chemical warfare. Even yeasts, themselves fungi, but unicellular fungi quite different from the filamentous forms we've been discussing, can form partnerships with mycelium. Yeast symbionts aid stress tolerance in some fungal species. These yeasts colonize the surfaces of hyphae or live within the mycelial mat, and they produce compounds that help the fungus tolerate environmental stresses. Some yeasts produce osmoprotectants, molecules that help cells maintain function under salt stress or drought. Others produce antioxidants that protect against oxidative damage. Still others produce heat shock proteins or other stress response molecules that the fungus can benefit from. In exchange, The yeasts gain access to the nutrients that the mycelium makes available through its decomposition or mycorrhizal activities. They live in the microenvironment that the mycelium creates, the moisture, the pH, the availability of specific nutrients that might be more favorable than the bulk soil environment. It's another partnership, another instance of multiple organisms integrating their metabolisms and stress responses into a more resilient collective. Sometimes the partnerships are between different filamentous fungi. Helper fungi boost decomposition in complex substrates like wood. Some fungi are specialized for breaking down cellulose, others for attacking lignin, still others for metabolizing particular compounds found in bark or heartwood. When multiple fungal species colonize the same piece of wood, they often decompose it faster and more completely than any single species could alone. Each fungus contributes its particular enzymatic capabilities, its particular tolerances for the chemical changes occurring as wood breaks down, and collectively they accomplish what individuals cannot. This isn't necessarily cooperation in the sense of organisms actively helping each other. Often it's more like complementarity, each fungus doing what it does best, with the byproducts or partial decomposition products of one species becoming the substrate for another. But the result is a more efficient, more thorough breakdown of complex materials, and all the fungi involved benefit from the increased availability of nutrients. Even more intimate partnerships exist in mycorrhizal contexts. Dual mycorrhizae share nutrients between fungal species and their shared plant hosts. It's not uncommon for a single plant to be colonized by multiple mycorrhizal fungi simultaneously, perhaps an arbuscular mycorrhizal fungus and an ectomycorrhizal fungus, or multiple species of the same type. These fungi might have different spatial distributions, colonizing different parts of the root system, or different functional specializations, one better at acquiring phosphorus, another better at nitrogen or water uptake. And these different mycorrhizal fungi can connect through the plant. Nutrients absorbed by one fungus and delivered to the plant can then flow from the plant to the other fungus. The plant becomes a mediator, a common partner that links different fungal networks. And because each fungus is itself connected to multiple plants, the result is a multi-layered network, plants connected to fungi, fungi connected to plants, creating a web of interactions where resources flow in complex patterns determined by the needs and availabilities across the entire system. In some cases, different fungal species growing on or near each other recognize each other as compatible and form anastomoses, hyphal fusions, directly linking their networks. This creates literal hybrid networks where the cytoplasm and resources of different fungal species can mix and flow between them. It blurs the line between individual and collective even further. Where does one organism end and another begin when their hyphae have fused, when their cytoplasms mix, when nutrients absorbed by one are transported through the body of another? But not all interactions are cooperative. Competition is constant in the soil environment, and fungi compete intensely with each other for space and resources. Yet even competition has structure, has rules, has mechanisms that prevent it from becoming mutually destructive. Chemical exclusion zones prevent conflict in dense fungal communities. When two fungal colonies growing through soil meet, they don't simply intermingle. Often they recognize each other as competitors, as non-self, and each produces higher concentrations of defensive compounds at the interface where they meet. The result is a boundary zone, an area of elevated antibiotic concentrations, sometimes visible as a distinct line where the two colonies meet but don't fuse. Each fungus is defending its territory, maintaining its exclusive access to the resources in its occupied zone. while respecting the equivalent boundary established by the competitor. It's a chemical demilitarized zone, established and maintained by mutual chemical aggression, ironically preventing the kind of direct physical combat that might damage both colonies severely. These exclusion zones can be remarkably stable, persisting for long periods, with the two fungal colonies growing in parallel, maintaining their boundary, neither advancing into the other's territory. It's a stalemate, but a functional one, allowing both fungi to coexist in proximity without constant, destructive warfare. The soil, looked at closely, is carved into territories, domains claimed and defended by chemical means, with mycelial networks of different species occupying adjacent zones in an uneasy but stable peace. And then there are the viruses. We don't often think of viruses in the context of fungal ecology, but they're there, pervasive and important. Many fungi host viruses, mycoviruses, that infect fungal cells and replicate within them. Some of these viruses are harmful, reducing fungal growth or reproduction, but others are beneficial or neutral. And in some remarkable cases, viral satellites suppress competitor viruses in a form of biological control at the viral level. A satellite virus is a virus that depends on another virus, a helper virus, to complete its replication cycle. It cannot replicate on its own, but when the helper virus is present, the satellite can use the helper's molecular machinery to reproduce. Some satellites are benign or even beneficial to their fungal host because they suppress the replication of the helper virus, which might be harmful. By competing with the helper for resources or interfering with its replication cycle, the satellite reduces the damage the helper virus causes to the fungus. This creates a three-way interaction: fungus, helper virus, and satellite virus, where the satellite is, in effect, protecting the fungus from the more harmful virus. The fungus hosts both viruses, but the presence of the satellite makes that hosting less costly. It's yet another layer of complexity, another instance where what appears to be a fungal network is actually a community of interacting organisms including multiple kingdoms of life: fungi, bacteria, yeasts, and viruses, all influencing each other's survival and success. The mycelium, then, is never alone. It is always colonized, always partnered, always embedded in networks of interaction that extend beyond the boundaries of its own hyphae. Bacterial endophytes fix nitrogen inside its cells. Actinomycetes coordinate antibiotic production in its vicinity. Yeast symbionts enhance stress tolerance. Helper fungi boost its ability to decompose complex substrates. Dual mycorrhizal partners share nutrients through common plant hosts. Chemical exclusion zones maintain territories between competing species. Satellite viruses suppress harmful viruses within its tissues. Every hyphal thread we've discussed is potentially host to bacteria. Every mycorrhizal association might involve multiple fungal species. Every competitive interaction is mediated by chemical signals and responses. Every fungal network exists within, and as part of, a larger community of microorganisms: the microbiome of the soil, the holobiont of the mycorrhiza, the meta-community of decomposers in the log. This perspective changes everything. We can't understand mycelium in isolation because it never exists in isolation. Its success depends on its bacterial partners providing nitrogen. Its survival depends on actinomycete allies enhancing antibiotic production. Its stress tolerance is augmented by yeast symbionts. Its ecological function is amplified by helper fungi with complementary capabilities. Its territorial stability depends on chemical exclusion zones negotiated with competitors. Even its viral infections are managed by satellite viruses. The mycelium is a community. The fungal network is a scaffold, a framework, an organizing structure, but the living, functional entity is the entire assemblage of organisms that inhabit it, partner with it, compete with it, coexist with it. The hyphae are highways traveled by bacteria. The hyphal walls are surfaces colonized by yeasts. The cytoplasm is a habitat for endophytes. The territory is a negotiated space bounded by chemical exclusion zones. We have been asking what mycelium is, how it grows, what it does. But perhaps the better question is what mycelium is part of. And the answer is everything. It is part of a hybrid network that includes bacteria and yeasts and other fungi and viruses. It is part of a nutrient cycling system that includes plants and decomposers and soil minerals. It is part of a communication network that links trees across forests. It is part of a competitive landscape where chemical boundaries are constantly negotiated and maintained. The mycelium never works alone. It cannot work alone. It is fundamentally, essentially, necessarily embedded in communities, in networks, in webs of interaction that make the distinction between individual and collective almost meaningless. Where does the fungus end and the bacteria begin when bacteria live inside fungal cells and provide essential nutrients? Where does one fungal network end and another begin when they've fused their hyphae and are sharing resources? Where does the mycelium end and the forest begin when mycorrhizal networks link every tree? These questions have no clean answers because nature doesn't respect the boundaries we draw. Life is interconnected, interdependent, intermingled. The mycelium is perhaps the ultimate expression of this truth, a network organism, a community as individual, a living testament to the power of connection, cooperation, and the blurred boundaries between self and other, between organism and ecosystem, between individual and collective. It never works alone. It was never meant to work alone. And in that collaboration, in that mixing of metabolisms and molecules and species, lies its strength, its resilience, its success across hundreds of millions of years. The mycelium, host and partner, competitor and cooperator, individual and community, all at once, all the time, in every thread, in every connection, in every moment of its existence. Allies, rivals, and hybrid networks. This is the true nature of mycelium, not singular, but plural, not isolated, but embedded, not alone, but always, inevitably, beautifully, part of something larger than itself. We have followed the mycelium from spore to network, from microscopic thread to vast underground web. We have seen it grow, communicate, defend, cooperate. But every story of life must eventually turn to the question of continuation, of legacy, of the next generation. How does mycelium ensure its survival beyond the lifespan of any individual network? How does it colonize new territories, spread to fresh substrates, persist across time and space? The answer lies in reproduction, in the production of spores, those microscopic capsules of potential that can travel on wind, in water, on the bodies of animals, carrying the genetic information of the parent mycelium to distant locations where new colonies can begin. This is preparation for the next generation. This is how mycelium expands beyond the limits of hyphal growth, how it achieves what its thread-like form alone cannot, movement across barriers, dispersal across distances, the colonization of the wider world. There are many strategies for reproduction in the fungal kingdom, many ways that mycelium produces the spores that will seed new colonies. Let's begin with perhaps the simplest and most direct. Many fungi reproduce asexually, creating spores that are genetic clones of the parent. For this, specialized structures develop. Conidiophores produce asexual spores called conidia. A conidiophore is a modified hypha, standing upright from the mycelial mat, often branched in characteristic patterns that differ between fungal species. At the tips of these branches, or sometimes along their length, spores bud off or form in chains. These conidia are not the product of sexual fusion. There's no mixing of genetic material from two parents, no meiosis, no recombination. They're produced mitotically, simple cell division, creating spores that are genetically identical to the parent mycelium. This allows for rapid reproduction when conditions are favorable. If a fungus has successfully colonized a substrate, if its particular genetic configuration is well suited to the current environment, why change? Asexual reproduction through conidia allows the fungus to produce vast numbers of spores quickly, all carrying the same successful genetic formula. The conidiophores can be elaborate structures. In some species, they're simple stalks with spores at the tip. In others, they branch like tiny trees, with spores forming on every branch. In still others, they form brush-like or vesicle-like arrangements that maximize spore production in minimal space. When viewed under magnification, these structures have their own architecture, their own elegance, purpose-built spore factories, each designed by evolution to maximize the production and dispersal of the next generation. Another asexual strategy involves different structures. Sporangiophores elevate sporangia above the substrate. A sporangiophore, like a conidiophore, is a modified hypha extending upward, but at its tip it forms a sporangium, a sac-like structure within which spores develop. These spores, called sporangiospores, form inside the protective sporangium, often in vast numbers. A single sporangium might contain thousands or tens of thousands of spores. When mature, the sporangium ruptures, releasing its spores in a burst or gradually as the sporangium wall degrades. The sporangiophore has lifted the sporangium away from the substrate surface, positioning it where air currents are stronger, where the spores have a better chance of being carried away rather than settling immediately back onto the already colonized material. Its elevation for dispersal, height achieved through the simple mechanism of a single hypha growing upward, swelling at the tip, and filling with spore-producing tissue. The sporangium wall itself is often designed for optimal dispersal. Some species have walls that are hydrophilic when dry, becoming sticky when wet so that water droplets can splash disperse the spores. Others have walls that dry to brittleness, shattering with the slightest disturbance to release spores as dry powder. Still others have walls that dissolve gradually, releasing spores slowly over time rather than all at once. Each strategy optimizes dispersal for the particular ecology of the species: water dispersed in wet environments, air dispersed in dry ones, gradual release where conditions are stable, and immediate mass dispersal is unnecessary. But not all fungal reproduction is asexual. Sexual reproduction, the fusion of genetic material from two compatible parents, occurs in most fungal species, and it produces structures and spores of remarkable sophistication. In the Basidiomycota, the division that includes mushrooms, bracket fungi, and many other familiar forms, sexual spores form on specialized cells called basidia. Basidia form on terminal hyphae in fruiting body tissues or sometimes directly on the mycelial surface. A basidium is a club-shaped cell where the magic of sexual reproduction culminates. Two compatible haploid nuclei, one from each parent, that have been coexisting in the hyphae finally fuse within the basidium, creating a brief diploid stage. This diploid nucleus immediately undergoes meiosis, the special cell division that produces 4 haploid nuclei, each genetically unique due to recombination and the random assortment of chromosomes. These 4 nuclei are the genetic seeds of the next generation. But they can't simply be released as nuclei. They need protection, need resources to survive until they land somewhere suitable and germinate. So the basidium develops 4 tiny projections called sterigmata, delicate stalks that extend from the basidium's surface. And at the tip of each sterigma, a spore begins to form. Sterigmata produce basidiospores, the sexual spores that will disperse and, if fortunate, germinate to start new mycelial colonies. Each basidiospore receives one of the 4 nuclei from the meiotic division, along with a package of cytoplasm, mitochondria, ribosomes, nutrients, everything needed to germinate and begin growth. The spores are typically small, often beautifully ornamented with ridges, spines, or other surface features that help them catch air currents or adhere to dispersal vectors. When mature, they're forcibly discharged from the sterigmata through an elegant mechanism involving water condensation and surface tension, launching the tiny spores into the air with enough force to carry them away from the basidium surface and into air currents that might carry them centimeters, meters, or even kilometers from their origin. The production of basidia and their spores typically occurs in massive numbers within fruiting bodies, the mushrooms, brackets, and crusts that are the visible manifestations of mycelial reproduction. A single mushroom might produce millions or billions of basidiospores, each one a genetic experiment, a unique combination of the parent genomes, each one capable of founding a new colony if it lands in a suitable location. The odds against any individual spore succeeding are astronomical, but the numbers are so vast that some will succeed, ensuring the continuation of the lineage. But before basidia can form, before spores can be produced, the fruiting body itself must develop. And this development begins deep within the mycelium, often triggered by environmental cues, changes in temperature, moisture, nutrient availability, or light. The first visible sign is often a small, dense aggregate of hyphae called a hyphal knot. Hyphal knots initiate fruiting bodies. These knots are regions where hyphae stop their exploratory growth and instead grow inward, toward each other, interweaving tightly. What was a loose network becomes, in that localized area, a dense ball of tissue. This is the primordium, the earliest stage of fruiting body development. Within this knot, cellular differentiation begins. Some hyphae will become the tough, protective outer skin of the fruiting body. Others will form the structural tissue of the stalk or cap. Still others will differentiate into the spore-producing layer, the gills or pores where basidia will eventually form. The hyphal knot grows, expands, differentiates, eventually breaking through the substrate surface to emerge as a recognizable fruiting body, a mushroom pushing up through soil, a bracket fungus projecting from bark, a puffball swelling on the forest floor. The mycelium has invested enormous resources into this structure, diverting nutrients from growth and maintenance to the construction of the reproductive body. It's a calculated risk, an investment in dispersal, a commitment to the next generation. And the return on that investment is millions of spores, each one carrying the mycelium's genetic legacy, each one ready to travel, to wait, to germinate when conditions align. Spores enable rapid colonization of new substrates. This is their function, their purpose. A spore landing on fresh substrate, perhaps a newly fallen tree, perhaps disturbed soil rich in organic matter, perhaps the roots of a young plant seeking mycorrhizal partners, can germinate within hours or days if conditions are right. A hypha emerges, begins exploring, branching, forming a new mycelial network. What might take weeks or months for a mycelium to reach through hyphal growth, extending from one tree to another across meters of distance, a spore can achieve in seconds of wind dispersal. Spores cross barriers that hyphae cannot. They traverse gaps, travel over inhospitable terrain, disperse across landscapes. They're the fungal kingdom's answer to seeds, though they're far smaller, far more numerous, and in many ways far more successful at dispersal. Some spores can remain viable for years, waiting in soil or on surfaces, dormant but ready, until the moment when moisture and temperature and substrate availability all align. Others remain viable for only days or weeks but are produced in such astronomical numbers that some will inevitably land where conditions are immediately favorable. Different strategies, but the same goal: get the genetic material to a new location where a new colony can establish, grow, and eventually produce its own spores to continue the cycle. This is reproduction at the fungal scale. Not the slow fusion and development of animal reproduction, not the seasonal cycles of plant flowering and fruiting, but something more continuous, more opportunistic, more driven by sheer numerical abundance. Conidiophores budding off countless asexual spores. Sporangiophores elevating sporangia that burst with thousands more. Basidia forming in the millions within fruiting bodies, each producing 4 basidia spores, each spore a potential founder of a new colony. Hyphal knots initiating the massive structures needed to produce and disperse these spores. And the spores themselves, tiny beyond imagining, carried on every breeze, settling on every surface, waiting for the chance to germinate and begin the cycle again. This is how mycelium transcends space, how it colonizes new territories, how it ensures that no matter what happens to any individual network, no matter if it's consumed by grazers, outcompete by rivals, destroyed by environmental catastrophe, the species continues. Because somewhere, some spore will land in favorable conditions. Some will germinate. Some will grow. And the mycelial network will spread again, weaving through substrate, forming connections, partnering with plants and bacteria, defending territory, communicating through its living web. The next generation is prepared, packaged in microscopic capsules, dispersed on wind and water in the bodies of animals. The mycelium's investment in reproduction ensures its persistence across time, its expansion across space. From spore to network to fruiting body to spore again, the cycle continues, has continued for hundreds of millions of years, will continue as long as there are substrates to colonize and ecosystems to support the fungal way of life. This is reproduction and expansion. This is the mycelium's reach extending beyond the limits of hyphal growth, crossing distances and barriers, finding new substrates, new partners, new opportunities. This is the preparation of the next generation, the ensuring of continuation, the investment in future that makes all life possible. The spores are flying, settling, waiting. And when they germinate, when conditions align, the mycelium will spread again, threading through substrate, weaving networks, connecting the world beneath our feet. The cycle continues. Always continues. Spore to hypha to network to fruiting body to spore. The ancient pattern, the fundamental strategy, the way of the fungus. Reproduction and expansion, ensuring that the mycelium, in some form, in some place, always persists. For hundreds of millions of years, mycelium has been solving engineering problems in the natural world. How to span gaps. How to penetrate resistant materials. How to create structures that are simultaneously lightweight and strong. how to form networks that are resilient to damage. Nature, through evolutionary time, has refined these solutions to remarkable precision. And now, humans are beginning to pay attention. We are looking at mycelium not just as an organism to study, but as a technology to harness. Nature is becoming technology, and mycelium is at the frontier of this transformation. From construction materials to medical scaffolds, from packaging to drug delivery systems, the properties that make mycelium successful in nature are being adapted to solve human problems in ways that are sustainable, biodegradable, and surprisingly elegant. Let's begin with materials, with structure, with the question of what mycelium can build. When fungal hyphae colonize a substrate, say, agricultural waste like straw or wood chips, they don't simply grow on the surface. They penetrate throughout the material, weaving between particles, binding them together with their hyphal networks. The result is a composite material, substrate particles held in a living matrix of fungal tissue. And if this colonization is allowed to proceed, if the mycelium is then killed and dried, through heat or desiccation, what remains is a stable material with remarkable properties. Mycelium forms biofoam-like composites that have captured the attention of materials scientists and engineers worldwide. These mycelial composites are incredibly lightweight. The hyphal network between substrate particles is mostly air, just thin-walled tubes creating a three-dimensional scaffold. The bulk density is low, comparable to expanded polystyrene foam or other synthetic foams used in packaging and insulation. But unlike synthetic foams derived from petroleum, mycelial composites are grown from agricultural waste, require minimal energy input during production, and are completely biodegradable at the end of their useful life. They can be composted, returning their materials to the soil, closing the loop in a way that synthetic materials simply cannot. But low weight alone isn't enough. The material must also have useful mechanical properties. And here, mycelium surprises us. The way mycelial composites respond to stress depends on the type of stress applied. When pulled, subjected to tension, the material exhibits strain hardening under tension. Strain hardening means that as the material is stretched, it becomes stiffer, more resistant to further deformation. This is unusual and valuable behavior. What's happening at the microscopic level is that the hyphal networks are being pulled taut. Initially, some hyphae are slack, some are loosely coiled, some are taking indirect paths through the material. But as tension increases, these hyphae straighten and align with the direction of stress. As more and more hyphae become load-bearing, the material's resistance to further stretching increases. It's a progressive stiffening, a mechanical response that helps the material resist tearing and distribute stress across a larger volume. This makes mycelial composites tougher than you might expect from such a lightweight material. But what about compression? When squeezed rather than pulled, mycelial composites behave differently. They behave like open-cell foam under compression. Open-cell foam, think of a kitchen sponge or the foam inside seat cushions, consists of a network of struts surrounding open spaces filled with air. When compressed, the foam deforms by the struts bending and buckling, and the air escaping from the cells. This gives a characteristic compression curve, initial ease of compression, then increasing resistance as the structure densifies. Mycelial composites show this same behavior. When compressed, the hyphal networks collapse, the air spaces between substrate particles and hyphae decrease, and the material densifies. It's soft initially, becoming harder as compression continues and the structure compacts. This makes mycelial composites useful for protective packaging, They can cushion impacts by absorbing energy through compression, just like traditional foam packaging, but with the environmental benefits of being biodegradable and grown from waste materials. Companies are already producing mycelium-based packaging materials, insulation panels, and even building blocks. Walls can be constructed from mycelium bricks grown in molds, lightweight and insulating, though not load-bearing in the same way as concrete or fired brick. Furniture has been prototyped and produced, using mycelium as the core material, sometimes with surface treatments to give durability and aesthetics. The potential applications are vast. Anywhere synthetic foams or lightweight composites are currently used, mycelium offers a biological alternative. But the applications extend far beyond construction and packaging. The biomedical field has taken particular interest in mycelium's properties, because the same features that make it good at binding substrate particles and creating lightweight structures also make it promising as a biomaterial for tissue engineering. The three-dimensional network of hyphae, with its interconnected spaces and fibrous structure, turns out to resemble something else at the microscopic scale: the extracellular matrix that surrounds cells and animal tissues. The extracellular matrix is the scaffolding that gives tissues their structure, that cells attach to and migrate through, that provides the physical context for cellular function. In tissue engineering, the attempt to grow replacement tissues or organs outside the body, one of the major challenges is creating a scaffold that cells can colonize, that supports their attachment and growth, that has the right mechanical properties and porosity. And it turns out that mycelium, with some processing and modification, can serve this function. Mycelial scaffolds mimic human extracellular matrix in important ways. The hyphal networks provide a fibrous structure, much like the collagen fibers of natural extracellular matrix. The spaces between hyphae allow for cell infiltration and the diffusion of nutrients and waste products. The material is porous, interconnected, three-dimensional, all properties that tissue engineering scaffolds need. And with appropriate treatment, perhaps chemical modification of the hyphal surface, or coating with proteins that cells recognize, mycelial scaffolds can be made biocompatible, suitable for contact with living tissue. Research has shown that these scaffolds support fibroblast attachment. Fibroblasts are the cells that produce and maintain the extracellular matrix and connective tissues. They're fundamental to wound healing, to tissue structure, to the integrity of skin and organs. When fibroblasts are seeded onto mycelial scaffolds in laboratory culture, they attach. Their surface receptors recognize sites on the mycelial material where they can bind. They spread out across the hyphal surfaces, extending processes, beginning to behave as they would on natural extracellular matrix. And crucially, they survive and thrive. The scaffolds promote cell viability. Cells attached to mycelial scaffolds remain alive, maintain their metabolic functions, and begin to proliferate, dividing to increase their numbers. This is essential. A scaffold that cells attach to but then die on would be useless. The mycelial material must not only provide a physical structure but also a biochemical environment that supports cellular life. The porosity allows nutrients from the culture medium to reach cells throughout the scaffold. The structure allows waste products to diffuse away. The material itself doesn't release toxic compounds or trigger overwhelming immune responses. With further development, such scaffolds might be used to grow skin grafts for burn victims, to engineer cartilage for joint repair, to create vascular grafts, or even to serve as frameworks for more complex tissue structures. The biodegradability of mycelium becomes an advantage here, as cells colonize the scaffold and produce their own extracellular matrix, the mycelial material can gradually degrade and be replaced by the natural tissue matrix, leaving behind fully biological tissue without any permanent synthetic implant. But the biomedical applications don't stop with scaffolds. Mycelium's porous structure and the ease with which it can be processed and shaped make it attractive for another critical application: drug delivery. The challenge in pharmaceutical medicine is often not just getting a drug into the body, but controlling where it goes and how fast it's released. Many drugs work best when released gradually over time, maintaining therapeutic concentrations without the peaks and valleys that come from taking discrete doses. Mycelial materials can be loaded with drugs, either by growing the mycelium in the presence of the drug compound so it absorbs into the hyphal tissue, or by soaking processed mycelium in drug solutions. Once loaded, the mycelium can be formed into whatever shape is needed, perhaps a small implantable pellet, perhaps a coating on a surgical site, perhaps a component of a wound dressing. The drug loads into the porous structure, held by physical adsorption or weak chemical interactions. And then, as the mycelium slowly degrades in the body's aqueous environment, as its hyphal walls break down and its structure dissolves, the drug is gradually released. This enables controlled release over hours, days, or even weeks, depending on the specific mycelial material used, the processing it received, and the biochemical environment where it's placed. The rate of release can be tuned by adjusting the density of the mycelial material, the degree of cross-linking in the hyphal walls, the size and shape of the delivery vehicle. This controlled release reduces the need for frequent dosing, improves patient compliance, maintains more stable drug levels, and can deliver drugs specifically to the site where they're needed rather than systemically throughout the body. For treatments requiring local drug delivery, fighting infection at a wound site, delivering chemotherapy directly to a tumor, providing anti-inflammatory drugs to damaged tissue, mycelium-based delivery systems offer advantages over traditional methods. The elegance of these applications lies in their biomimicry. We're not trying to create something entirely new and synthetic. We're recognizing that mycelium has already solved problems we're trying to solve, creating lightweight composites, forming three-dimensional scaffolds, managing the absorption and release of compounds. Evolution has been working on these problems for hundreds of millions of years. The solutions are already there, refined and tested at scales and timescales we cannot match in laboratories. We just need to understand them well enough to adapt them to our purposes. And there's a sustainability aspect that cannot be ignored. Traditional materials science relies heavily on petroleum derivatives, on energy-intensive manufacturing processes, on materials that persist in the environment for centuries after their useful life ends. Mycelial materials are grown, not manufactured in the traditional sense. They can be produced from agricultural and forestry waste, materials that would otherwise be burned or landfilled. The growth process requires some energy for climate control and sterilization, but far less than polymer synthesis or foam production. And at the end of life, these materials biodegrade. They can be composted. They return to the earth as nutrients, completing a cycle rather than accumulating as persistent waste. This is circular economy thinking at its most literal. Materials that come from biological waste serve a useful function and then return to biological cycles. It's the opposite of the linear take-make-dispose model that has characterized industrial production for the past century. And mycelium, with its rapid growth, its ability to colonize waste materials, and its useful material properties, sits at the center of this emerging paradigm. we are still in the early stages of understanding and utilizing mycelial materials. The science of controlling growth conditions to optimize material properties is still developing. The engineering of surface treatments and modifications to expand applications is ongoing. The regulatory pathways for medical applications are being navigated. But the potential is clear and vast. Nature is becoming technology. The same hyphal networks that decompose logs in the forest, that partner with tree roots, that communicate and defend and reproduce, these networks can be harnessed to create foams and scaffolds in drug delivery systems. The material science that evolution performed over geological time is being translated into human applications, sustainably, elegantly, with minimal environmental cost. Mycelium forms biofoam-like composites, lightweight, strong, biodegradable. It exhibits strain hardening under tension and behaves like open-cell foam under compression, mechanical properties useful for packaging and protection. Its scaffolds mimic the extracellular matrix, fibrous, porous, three-dimensional. It supports fibroblast attachment and promotes cell viability, the foundations of tissue engineering. It loads drugs for controlled release, opening pathways for better pharmaceutical delivery. From forest floor to factory floor, from decomposer to material, from ancient organism to cutting-edge technology. The mycelium continues to surprise us, continues to offer solutions, continues to demonstrate that the wisdom embedded in natural systems, refined over vast stretches of evolutionary time, remains relevant and valuable as we search for sustainable ways forward. Nature becomes technology. And in this transformation, mycelium leads the way, threading its ancient networks into the future of materials, medicine, and human innovation. The same threads that bind substrate particles in the forest are binding particles in packaging materials. The same scaffolds that support bacterial endophytes are supporting human cells and culture. The same porosity that allows cytoplasmic streaming allows drug diffusion. The mycelium, once again, proves itself adaptable, useful, essential. Not just to forest ecosystems, not just to plant partners, but now to human endeavors as well. Nature's engineering, billions of years in development, available for those wise enough to learn from it, sustainable enough to use without destroying, elegant enough to inspire wonder even as it solves practical problems. This is mycelium as technology. This is nature's wisdom applied. This is the future growing, quite literally, from the threads of the past. We have traveled from the microscopic to the planetary, from single hyphae to forest-spanning networks, from ancient evolutionary origins to cutting-edge biotechnology. But now, as we approach the end of our journey through the world of mycelium, let us turn our gaze upward. Beyond the atmosphere. Beyond Earth's gravity well. To the Moon, to Mars, to the orbital stations and distant outposts where humanity dreams of establishing itself among the stars. And ask a question that might seem fantastical, but is being seriously pursued by scientists and engineers. Could mycelium, these fungal threads that decompose logs and partner with roots, could mycelium build our homes in space? Could the same organism that has shaped terrestrial ecosystems for hundreds of millions of years become the foundation for extraterrestrial architecture? The answer, remarkably, appears to be yes. This is architecture beyond Earth. This is the audacious vision of mycelium not just sustaining life on this planet, but enabling it to spread to others. The fundamental challenge of space architecture is mass. Launching material from Earth's surface into orbit, let alone to the Moon or Mars, requires enormous amounts of energy and is staggeringly expensive. Every kilogram of payload costs thousands of dollars to lift into space. Traditional construction materials, steel, concrete, even wood, are heavy. A habitat capable of protecting astronauts from radiation, from temperature extremes, from micrometeorite impacts, built from conventional materials, would have a mass measured in tons. Getting that mass off Earth is prohibitively difficult and expensive, which is why current space habitats are cramped, minimal, optimized for every gram. But what if instead of launching finished structures, we launched something smaller? Something that could grow into a full-scale habitat once it arrived at its destination? This is where mycelium enters the picture, because one of its most remarkable properties is its ability to produce large volumes of structured material from minimal starting mass. Mycelium can self-assemble habitats, transforming compact packages of dormant fungal tissue and substrate into full-sized structures through biological growth. The concept is elegant in its simplicity. A habitat module would be launched in a dormant, compressed state, essentially freeze-dried mycelium combined with a substrate it can grow through, perhaps held in a thin, inflatable shell. This package would be flat-packed or tightly compressed, taking up minimal space in the spacecraft. The mass would be a fraction of what a conventional habitat would weigh, perhaps hundreds of kilograms instead of many tons. Flat-packed structures activate with water. Upon arrival at the destination, a lunar base site, a Martian settlement location, an orbital station, water would be added to the dormant package. This is all it takes to wake the mycelium. Water triggers germination, rehydrates the dormant hyphae, activates the cellular machinery that has been waiting, sometimes for years if necessary, in suspended animation. The mycelium begins to grow, hyphae extending from their dormant state, branching, exploring the substrate they're packaged with. Within hours, growth is visible. Within days, the mycelium has colonized the entire substrate, binding it together, creating a composite material. But simply growing mycelium through a pile of substrate wouldn't create a useful habitat. Structure is needed, shape, form that can be inhabited. This is where another elegant design element comes in. The dormant mycelium package includes an inflatable scaffold, a thin polymer or fabric structure that can be inflated to the desired shape of the habitat. Think of it as a balloon, but one designed to be the template for biological growth. The mycelium grows around inflatable scaffolds, using them as formwork, as molds that define the final architecture. The inflatable expands, creating the three-dimensional form, perhaps a dome, perhaps a cylinder, perhaps a more complex shape designed for specific functions. And as the mycelium grows, it colonizes the substrate surrounding and adhering to this inflatable form, binding it all together, creating a rigid, self-supporting structure that conforms to the scaffold shape. The scaffold can remain as an internal layer, providing an airtight barrier, while the mycelial composite provides structural support, insulation, and protection from external hazards. The advantages of this approach are profound. The launch mass is reduced dramatically compared to conventional construction. You're launching mostly dormant biological material and water, not finished habitat modules. The volume taken up during launch is minimal because everything is compressed or deflated. Only at the destination, using locally available water if possible, extracted from ice on the Moon or Mars, does the habitat expand to its full size. It's like planting a seed and watching a building grow. And the material properties of the grown structure are well suited to the space environment. One of the greatest hazards in space is radiation, cosmic rays and solar particle events that can damage DNA, increase cancer risk, and cause acute radiation sickness during major events. On Earth, we're protected by our atmosphere and magnetic field. In space, habitats must provide this shielding themselves. Conventional materials require significant thickness to provide adequate radiation protection, meters of regolith, thick metal walls, or massive water tanks. But mycelial structures can be designed with integrated radiation shielding. The mycelium itself provides some protection. Organic material absorbs radiation better than vacuum. But the real advantage comes from the architecture. Mycelial walls can be grown with spaces or channels designed to be filled with water ice. The habitat provides radiation shielding with ice layers incorporated into its walls. Water is excellent at absorbing radiation. Hydrogen atoms are particularly effective at capturing neutrons and moderating charged particles. By growing a mycelial structure with cavities that are then filled with ice, you create a habitat with effective radiation shielding without requiring enormous additional mass at launch. The ice can potentially be sourced locally, from lunar polar craters, from Martian permafrost, further reducing what must be brought from Earth. But living in a sealed habitat for months or years presents another challenge: life support. Air must be recycled, oxygen replenished, carbon dioxide removed. Food production would ideally occur locally rather than relying entirely on resupply from Earth. Here again, the biological nature of mycelial habitats offers advantages. The system can be designed as a symbiotic ecology, not just a sterile structure. Cyanobacteria supply oxygen and nutrients to the habitat ecosystem. Cyanobacteria are photosynthetic bacteria, sometimes called blue-green algae, that can convert carbon dioxide and water into oxygen and organic matter using light energy, just like plants but generally faster and in more compact systems. Cultures of cyanobacteria can be integrated into the habitat's life support system, consuming the carbon dioxide exhaled by astronauts and producing oxygen. But they can do more than that. The organic matter they produce, their biomass, can feed other organisms in the habitat ecology, including the mycelium itself. The mycelium in the habitat walls doesn't have to remain dormant after initial construction. It can be kept alive, growing slowly, maintained as part of a living building. And if fed with nutrients, perhaps from cyanobacterial cultures, perhaps from astronaut waste products being recycled, it can continue to produce enzymes that help break down waste, can maintain and repair the structure, can even produce useful compounds like vitamins or pharmaceuticals if genetically engineered for that purpose. The habitat becomes a living system, a symbiotic community of cyanobacteria, fungi, and humans, each supporting the others. The speed of construction is another remarkable advantage. Traditional construction in space is slow. Every component must be carefully assembled in vacuum or reduced gravity, often requiring spacewalks, always requiring time and precision. But biological growth, once initiated, proceeds on its own schedule. Growth completes in days for mycelial habitats. From the moment water activates the dormant mycelium to the point where the structure is fully colonized and rigid enough to be functional might take only a week or two, depending on the species of fungus chosen, the temperature maintained, and the size of the structure. This rapid construction timeline means habitats can be grown quickly upon arrival at a destination, reducing the time astronauts must spend in cramped spacecraft or temporary shelters. It means that if a habitat is damaged, by micrometeorite impact, by equipment failure, by whatever unforeseen challenges arise, replacement habitats can be grown quickly rather than waiting months or years for resupply from Earth. And the shape of these habitats need not be limited to simple domes or cylinders. Shapes can be guided by nutrient placement within the structure. Just as mycelium responds to nutrient gradients on Earth, growing densely where resources are abundant and sparsely where they're scarce, the same principles can be applied to controlling growth in space habitats. By placing higher nutrient concentrations in areas where thicker walls or denser material is desired, and lower concentrations where openings or thinner sections are needed, the growth can be directed, sculpted, guided to create complex architectures without requiring complex molds or scaffolds. Windows might be created by simply not placing substrate in those locations, allowing the inflatable scaffold to remain exposed. Internal partitions might be grown by placing vertical planes of substrate within the larger structure. Varying densities for different functional zones, thicker, denser walls on the exterior for protection, lighter, more porous internal structures for insulation or acoustic dampening, all can be achieved through careful design of nutrient and substrate distribution before growth begins. Compared to traditional space construction materials, mycelium holds its own or exceeds them in key metrics. Mycelium outperforms concrete and wood off-planet in several ways. Concrete requires water, aggregates, and cement, all of which must either be launched from Earth or manufactured from local resources using energy-intensive processes. Wood must be grown in controlled agricultural settings, requiring significant life support infrastructure, time, and resources. But mycelium grows from agricultural waste that can be compressed and transported efficiently, requires only water to activate, grows rapidly with minimal energy input beyond maintaining temperature, and creates a material with useful structural and insulative properties. The compressive strength of densely grown mycelial composites can rival lightweight concrete. The insulative properties are excellent. The porous structure traps air, providing thermal resistance important in the extreme temperature swings of airless environments. The material is self-healing in a sense. If kept alive, minor damage can be repaired by continued growth. And if a section must be replaced, new mycelial material can be grown and grafted onto the existing structure, something impossible with conventional construction materials. This self-repair capability is crucial for long-duration missions. Habitats will face where micrometeorite impacts, thermal cycling, mechanical stress. The ability of mycelial structures to maintain themselves, to repair minor damage through biological growth, to be patched with newly grown material that integrates seamlessly with the existing structure. This enables self-repairing surfaces that reduce maintenance burden and increase the longevity of space infrastructure. And the applications extend beyond habitats themselves. Mycelium supports space filtration and mining operations in proposed systems. The same enzymes that break down organic matter on Earth can be harnessed to process waste in space habitats. Sewage, food scraps, packaging materials can be fed to mycelium cultures that break them down and recycle the nutrients. Water filtration through mycelial biofilters could remove contaminants and pathogens, providing an additional layer of safety in life support systems. Even in situ resource utilization, the extraction and processing of materials from asteroid, lunar, or Martian sources, might benefit from mycelial biotechnology. Some fungi can solubilize minerals, extracting metals from ore through bioleaching processes. Others can survive in extreme conditions, high radiation, temperature extremes, low water availability, that might exist on other worlds. Genetically engineered fungal strains might be designed to extract specific elements from regolith, to bind toxic perchlorates found in Martian soil, to produce useful materials from the raw resources of whatever world we're attempting to colonize. This is not distant, speculative science fiction. NASA and other space agencies are actively researching these concepts. Prototype mycelial structures have been grown and tested. The fundamental biology is understood. The engineering challenges are being worked through: how to keep mycelium dormant during years-long transits, how to ensure reliable growth in low gravity and high radiation, how to integrate biological structures with the electronic and mechanical systems that habitats require. But the vision is clear and increasingly achievable. Mycelium may build humanity's future beyond Earth. The same organism that enabled plants to colonize land hundreds of millions of years ago, that built the forests and grasslands that cover our planet, that has been decomposer, symbiont, communicator, engineer throughout the history of terrestrial life. This organism might be the key to humanity's next great expansion. Not through metal and glass and concrete, but through living structures grown from simple materials, self-assembling, self-repairing, integrated into closed-loop ecological systems that can sustain human life far from Earth's protective embrace. Picture a lunar base, domes of mycelial composite glowing softly with interior light, their walls thick with ice shielding against solar storms, cyanobacteria cultures producing oxygen in transparent sections, mycelial networks quietly recycling waste and maintaining structure. Picture Martian settlements, hemispherical habitats grown in weeks from compressed packages, their shapes guided by nutrient gradients, their interiors partitioned into living quarters, laboratories, greenhouses, all held together by living fungal architecture that adapts and repairs itself as needed. Picture orbital stations, asteroid mining outposts, generation ships on journeys to other stars, all using mycelial technology to create livable space from minimal launched mass, to recycle and sustain, to provide the foundation for human presence wherever we dare to go. The threads that bind the Earth might someday bind our outposts on other worlds, weaving humanity into the cosmos through the same networks that have woven ecosystems on this planet for eons. This is the vision. This is the potential. Ancient organism, future technology. Earthly decomposer, cosmic constructor. The mycelium, which has worked in darkness and in silence beneath our feet for all of human history, might someday grow in the harsh light of alien suns, might spread through the regolith of other worlds, might shelter and sustain our species as it reaches beyond its homeworld. We end with awe, with the recognition that these microscopic threads, these simple tubes of chitin and cytoplasm, these networks of seemingly passive growth, they contain within them solutions to problems we're only beginning to face. They offer pathways forward that are sustainable, elegant, biological rather than purely mechanical. They demonstrate that the wisdom embedded in living systems, refined through evolutionary time, remains our greatest resource as we confront the challenges of our future, whether those challenges are on Earth or among the stars. The mycelium, patient and persistent, ancient and adaptable, decomposer and builder, has been preparing for this role for hundreds of millions of years. Now, finally, we are beginning to understand what it has to offer. And what it offers is nothing less than the architecture of our expansion into the cosmos, grown from dormant packages, activated with water, shaped by nutrients, living and self-repairing, sheltering humanity in environments that would otherwise be utterly hostile to our existence. From spore to space station, from forest floor to Martian surface, from the invisible beginning to the cosmic future. This is mycelium. This is what these threads can build. This is where the networks might extend, beyond Earth, beyond the limits we have known, into the vast possibilities that wait among the stars. The mycelium grows. It has always grown. And now, perhaps, it will grow in places life has never been, carrying with it the partnership between human and fungus, between technology and biology, between Earth's ancient wisdom and humanity's endless reaching toward new horizons. This is the architecture beyond Earth. This is the future, growing in the darkness, waiting for water, ready to expand and shelter and sustain. The same organism that enabled the forests enables, potentially, the stars. And in that potential lies both humility and hope, humility before the elegant solutions that life has already discovered, hope that by learning from and partnering with living systems, we might build a future that is sustainable, resilient, and truly alive, wherever in the cosmos we find ourselves. The mycelium waits. The future approaches. And the threads that bind might someday stretch from world to world, network to network, connecting not just trees in a forest, but outposts across the solar system, all held together by the ancient, patient, remarkable architecture of fungal filaments. Growing. Always growing. Into the future. Into the stars. Into possibilities we are only beginning to imagine. This is mycelium. This is what it can build. This is where we might go together, human and fungus, Earth and cosmos, ancient partnership reaching toward an extraordinary future. The journey ends here, but the growth continues. Always continues. Onward, upward, outward, the mycelium extends, and we extend with it, into the vast unknown, building our future one hyphal thread at a time.